Hey guys, if you're here on the channel, go ahead and leave a comment so that I can see if they're working. While I'm on here, I'll be able to see them. All right, it's 3 o'clock, so we're going to go ahead and get started. And if anybody else wants to join in, they can. So I'm just going to start in chapter 8 and kind of do like a short run through of it. And if there's anything that I can explain on the board in a diagram, I'm going to do that. And if you need me to stop and answer some kind of question, the chat should be open. If it's not, send me a message and group me because I have it here. Okay, Madison, I just saw your comment. And Erica, I'm going to see if I can send out another link real quick. Let's see if I can share it. Okay, Madison, how did you get on it? Which link did you follow? The last one. Okay. Let's try. All right, sorry guys, I'm trying to get this figured out. Okay. Sorry, we're trying to figure this out. Okay, so I think the first link I sent you guys is actually the link to my half of the video where I'm live streaming. So let me figure out. It gives me an option to share, but not to group me. Let me see if I can email it to myself. Okay, I'm gonna try this real quick. Okay, I'm gonna go to my YouTube app and see if I can just search the title and maybe that'll do it. Mm. Okay, I'm gonna log into let's see. Yeah, I'm on my computer and it pulls it up there, but it doesn't pull it up on my phone either. I'm gonna try and get in GroupMe on the computer and see if it will let me send it through here. You might have to be on a laptop for it to work. Oh, that's so weird seeing myself, but on my phone, but on here. And it's live. Okay, I see Jasmine's on here. Yay, that means the chat's working. Okay. All right, good deal. I want to wait and see if Erica can get in and maybe wait for a couple more minutes and see if anybody else wants to get on. Can you guys hear me okay? I feel like I'm loud, but that's just me. Okay. Yay, it's working. All right. Hey, Erica. All right, we're waiting for Callie to see if she can get in. It's not wanting to work on her phone either. Yeah, I totally would have done Zoom, but then the people that couldn't make it, that sucks because then it's just gone. So hopefully when we finish this, it'll stay archived and somebody can go back and watch it this afternoon if they want to. If you guys have any questions like from the get-go, you can go ahead and type those in the comments, and that way I'll know when to stop, or you can just wait until we get to that point if you want to. All right, sorry, we're still waiting for some people. Oh Lord. I don't know if it would pick up under my actual name or the Learn Me LPN. I think it's Learn Me LPN. Let me see if I can see from here. Okay, it should be. If it's not, then it's just my name. Okay, good deal. Okay. I think it's just Callie that's left right now, but it's her internet service because she's in Como and it's also raining. So we're waiting. Okay, she says she's gonna try and restart her Wi-Fi and try and get on so we can go ahead and get started. Um, thank y'all for waiting. Okay, so we'll start in the muscular system and we can just start at the beginning. So under muscle tissue, she's going to want us to know the differences between skeletal, cardiac, and smooth. And there's some printouts that I put on the website too. There's like 2 or 3 things that you need to know about each of these, and it doesn't get super serious into in depth until later. But basically, she wants you to know that skeletal muscle is striated and it's voluntary. So that means you're going to see kind of stripes in it if you're looking under a microscope, meaning striated. It's voluntary because you control that, and these are going to be attached to your bones. So skeletal muscle, know those. And sometimes the threads inside of each muscle cell are called muscle fibers. I'm not sure how much she's going to want us to know about it other than bolded terms for sure. So even if you can't tie it together just yet, at least know what the terminology is. So the cardiac muscle is going to be like the most part of your heart, and this is a hint. It's literally your heart muscle, cardiac meaning heart. So the fibers of these are cylindrical. I'm having a really hard time pronouncing those, but these branch out and kind of merge, and so these are more like a woven basket. So if you're thinking about your heart and it's having to pump blood from your chest all the way down to your tippy toes, it's going to be really strong. It's going to be reinforced in every other way so that it's strong enough to push out super, super far and then for it to come back up too. So this is going to be like a woven basket. It's just making sure all of its bases are covered so that it's able to pump it where it needs to go. So it's saying that under a microscope, they have dark staining bands that are cross sections of intercalated discs. And this is where the plasma membranes of adjacent cardiac fibers connect to one another. So that's basically what this is talking about. This is involuntary. So all day long, our heart's beating. We do not have to tell our chest to do this for us, thank God, because it'd be exhausting. And so this is an example of structure fits function. So the interconnected nature of cardiac muscle fibers help the tissue to contract as a unit and increase efficiency of the heart muscles in pumping blood. So, like I said, you're going to be thankful that they're woven together and built this way because it's going from your chest to your toes and even the teeny tiny little veins that are in your eyeballs. I mean, it's got to be able to push it through every single piece of your body. So we're going to jump over to smooth muscle, and these are tapered at each end and have a single nucleus. They lack cross stripes, so if you're looking at these under a microscope, you're not going to see that. And they're sometimes called non-striated muscle fibers. So they have a smooth, even appearance when viewed through a microscope, and these are involuntary as well. So these are going to be in your hollow organs, the walls of your blood vessels, internal organs, such as your gut, urethra, ureters, and this is visceral muscles. So we cannot control these either. Even though we're swallowing when we eat, we're not telling the muscles to pull the food down all the way through the alimentary canal. Also, this was one of the pop quiz questions that I had on the website as well, where peristalsis is the movement of the smooth muscle inside of your body to pull the food down and to digest it, and we don't control that. So that smooth muscle, that's something that's going to take care of itself. We don't have to worry about it. That also ties into parasympathetic and sympathetic nervous systems. So if you're at peace, you're at rest, your peristalsis is going to be working. You're going to be using a lot of smooth muscle during parasympathetic because you're relaxed and you're not having to worry about fighting, worry about running from something. So you're going to be able to digest things a lot quicker. And this is smooth muscle that allows us to do that. So it's involuntary and it's non-striated. So if we go down to muscle organs under the structure of skeletal muscle, skeletal muscle is an organ composed mainly of skeletal muscle fibers and connective tissue. And if you remember connective tissue we talked about in the last couple chapters, that's like one of the most predominant in your body. This is what keeps blood together, that kind of like sticky, gluey feel. And this is essentially kind of what holds you together, I would say. So fibrous connective tissue wraps around each individual muscle fiber and continues as it wraps around each group of muscle fibers. And these are called fascicles. So it forms a wrapper around the entire muscle organ, and fascia is the loose connective tissue outside of the muscle organs that forms a sticky, flexible packing material between muscles, bones, and skin. So in the study guide that I posted, there was a link for an autopsy. I don't know about y'all, but I like watching that kind of stuff. It's pretty cool. You can see, and if you've ever taken an A&P class where you've had to do a dissection, if you open up your chest and you're looking at your organs, you're going to have to go through this kind of like film before you can get to them. And it's just, that's what this is. That's what a fascia is. It's just a protective, like a plastic wrap kind of deal. Like if you're going to want to seal your food up before you put it in the refrigerator to keep it safe, that's kind of what this is, and it's over your organs. So I'm going to jump over to 153. So most skeletal muscles attach to two bones and have a movable joint between them. Some of these words start to run together to me, and it's easier for me to be able to draw. If it's confusing to you guys, I don't have to do that, but it's easier for me to explain it sometimes if I can do that. So on 153, when it's talking about skeletal muscles, you're going to involve your skeleton, you're going to involve your muscles. So you're going to have one bone and another bone, and then this is going to be your joint. So skeletal muscle is going to connect the two bones together, and this is what's going to be able to allow you to move them. So muscle's attachment to a more stationary bone is called its origin, and the attachment to the movable bone is called the muscle's insertion. So if this is we'll say I'm laying down in the bed. This is my body up here. So this is going to be my thigh, and this is going to be the bottom half of my leg, and then here's my feet down here. Terrible at drawing. So sorry, but I hope you can keep up. So the more stationary bone is going to be like more towards the center of your body. That's how I'm looking at it. So this part would be my origin. This is my joint right here. And this would be, what was the word? Insertion. Okay, so if that makes sense, the origin is going to be what is more stationary, and stationary meaning it's going to stay, it's more a positioned area. So when I think origin, I think I originated in Mississippi. This is where I've lived at. This is where I've been at. I'm staying here. And then insertion is going to be the moving part. So that'd be someone who moves from their origin. So the joint is going to connect the bones. This muscle is going to connect two bones. And then your origin is your more stationary. It's going to stay. It's your more strong point. And then your insertion is going to be the part that it pulls on. So the rest of the muscle, all of it except its two ends, is called the body of the muscle. So jumping down, tendons anchor muscles firmly to bones and are made of dense, fibrous connective tissue that extends from the muscle wrappers that we described earlier. So when I think tendons, hopefully everybody in here has been hunting at some point in their life. But if you've ever had to skin a deer, or if you've been in an A&P class, you'll see the like white kind of fiber things between a muscle. You'd even see them if you had bought a steak, the little white pieces between it. And those are like tendons. That's kind of what gives that pull feel. So these don't tear away from bone very easily, which means if you are in sports and you pull something, you pull a tendon, you injure a tendon, it's going to take a lot longer for you to bounce back from that. I'm going to take a break just a second and see if everybody's on here. All right. Checking on Callie. Okay, so we're going to jump down to bursae. These next few things are mostly memorization for the bolded terms. So bursae or bursae is between the tendons and the bones beneath them. And we did talk about this before. It's just a fluid-filled sac. And the small connective tissue sacs are lined with synovial membrane and filled with synovial fluid. So basically, this is what's going to keep your knee from giving out or your elbow from giving out. So these are found in joints. So elderly people who have issues with their joints may not have as much of this. So it's just a fluid that kind of keeps it lubricated. And if you think about your car, it's not going to run well if you don't put oil in it. It's not going to keep up as well if you don't maintain it. So we need this to be able to maintain the way that we are going. Okay, so let's see. It's a flexible cushion and reduces friction and makes it easier for tendon to slide over bone when the tendon's muscle shortens. So when you are working these muscles, it's just going to keep it lubricated. So now we're going to bump down to tendon sheaths. These enclose some tendons, and because these tube-shaped structures are also lined with synovial membrane, they're moistened with synovial fluid, and like bursae, they facilitate movement. So just associate bursae with tendon sheaths. This is all a point of lubrication. So does anybody have any questions so far? Am I going too deep into it? Not going deep enough into it? A lot of it, she may not really go super in depth, but sometimes it helps to know the why behind it for it to make sense. If you have any questions, put them in the comments. I can see it next to the video, and then I can stop and cover that. Okay, thank you, Jasmine. We're going to bump down to muscle fibers, the structure of muscle fibers. So skeletal muscle tissue is consisted of elongated contractile cells or muscle fibers that look like long tube-like cylinders. So you can see these on 153. Basically, the way that our body's built, we have different kinds of cells that stack. Our muscles are going to stack as well, and these specific muscle fibers have a specific shape to them. And so they're going to be kind of tube-like. Okay, so 153, the top of 153, we have that, but we'll just say that these are muscle fibers. And again, y'all, please excuse my terrible drawing right now. So they're connected, or flexible connective tissue wrappings hold them together in parallel groups and allow individual muscle fibers to pull them together in the same direction as a team. So if your muscles were not working together, it's going to be pretty chaotic. Can you explain tendon sheath again? Yes. Okay, so tendon sheaths enclose some tendons. These are tube-like structures that are also lined with synovial membrane and are moistened with synovial fluid. So like bursae, they facilitate body movement. So that's literally the only paragraph about that in the entire area. But basically, the tendon sheath, if we're looking at tendons, let's see if I can explain it in a picture. All right, so this is a muscle, and we'll say like this is a joint. No, I'm sorry. Joint. Okay. All right, so we have two bones connected with a muscle that's stretching over a joint. There's fluid-filled sacs within the edges of the joint, and these are the bursae. So if these have lubrication, we also want lubrication for the rest of it. So the tendons work with muscles. The tendon is like kind of placed in here. This is where we were talking about the steaks, or if you'd ever gutted a deer before you process meat, you're going to see the little white fibers throughout it. Well, these tendons have sheaths around them that help to keep them moistened so that they're not kind of burning out, if that makes any sense. So if like you have a belt, like a fan belt or something in your car, sometimes if you've had them for a long time, they're going to get hot and they'll burn out and they'll kind of snap. Not necessarily the case with this, but it's good to have the fluid. So a tendon sheath, when we talk about sheaths later, it's just a kind of like a jacket for it, just a little something to give it a cushion. So these tendon sheaths are what also lubricate these. These are so they're lined with synovial membrane and moistened with synovial fluid. So these are kind of like what we had talked about before in the past couple chapters with the fluid and lubrication. So does that make any sense to you? Okay, good deal. All right. So let's see. All right, so we talked about the muscles moving together as a team. So each skeletal muscle fiber has a unique cytoskeleton structure. The fiber's internal framework is organized into many long cylinders, each made up of two kinds of thread-like microfilaments, a thick and thin myofilaments. The thick myofilaments are formed from a protein called myosin, and the thin are composed of a protein called actin. So, y'all, please don't make fun of me the way I remember this stuff. So the thick and thin myofilaments, the thick one is formed by myosin. So that's the way that I remembered it. Oh my, she's thick. So I know that myosin is from a thick protein, or thick myofilaments are from a protein called myosin. And then so if we know that, we know the opposite. The second one is going to be the thin, and that's actin. It's a protein. So she's not really wanting us to get that in depth with us. She just wants us to know that in case we have to match that in a multiple choice question. So each shaft-like myosin molecule has a head that sticks out towards the actin molecule. And these are in the diagram on 153. So the proteins are kind of what are holding this together, and it's broken down into teeny tiny. Trying to find a picture we just had. It might not be in this chapter. It might have been in the older one. I'm going to see it. Might have been. No. It's similar to the nervous system. That's why I'm getting that confused. Okay. Skeletal muscle tissues consist of elongated contractile cells or muscle fibers that look like long tube-like cylinders, and their flexible connective tissue wrappings hold them together in parallel groups, allowing individual muscle fibers to pull together in the same direction as a team. So if you're looking at the diagram on 153, the pink and blue diagram that's on here is going to explain the thick myofilament and the thin one. So myosin and actin, these are what's going to be found in the sarcomere, and then it's going to come back up to muscle fiber. And this is basically, they're just breaking this muscle down into the smallest form that it's in. And these myosin and actin filaments are going to be what kind of hold it together and allow it to work together. So if you're thinking of maybe like a rowboat, where people compete and there's a bunch of people in a really long boat and they all have the paddles and they're going together. This is kind of what I think of when I see this. So they're literally just thicker and literally thinner pieces that are going to help to move this train together at one time, because if everything's pulling against each other, you're not moving anywhere. And then that kind of comes into play also later when we talk about things like Parkinson's, the things that are affected with that. Let's see. Okay, so now let's jump into figure 8-3. Where in the world does it say that at? Okay, which is the one at the top of 153. So it says, think of sarcomere as the basic functional or contractile unit of the skeletal muscle. The submicroscopic structure of sarcomere consists of numerous thick and thin myofilaments arranged so that when viewed under a microscope, dark and light stripes or cross striations are seen. The repeating unit stripes are separated from each other by dark bands called Z lines or Z discs. And this is the connecting of the thin myofilament. It's super hard to explain this in just talking about it, but the Z lines are kind of connections in like a chain link fence, if that makes any sense. So the way that it's showing us in the picture, let's see if I can do it like this. Okay, so there's connections here. This is deep, deep, deep inside of the muscle. The pink line itself, well, it's going to be black here, but these long lines themselves are the actin or the thin myofilament. So, and then these connecting chains right here in the middle are the Z lines that they're talking about. So it's holding the chain link fence together, if that makes any sense. Is this confusing y'all more? I hope it's not. It's hard to explain it in a diagram if I'm not like sitting next to you to show you, but I'm trying. Give me just a second. Okay. So now we're over on 154. I'm going to kind of jump around to, there's a sentence right above contraction of muscle fibers. It's a purple title. And it says, when a muscle is relaxed, the sarcomeres remain at their resting length and the myofilaments maintain their resting position. So the sarcomeres that it's talking about is the basic functional or contractile unit of skeletal muscle. And I'm trying to see if you can find it. Yeah. Okay, so all of this collectively is the sarcomere. And so in that sentence, it's telling us that when you're relaxing, it maintains at its resting length and the filaments maintain their resting positions. So I'm going to go down to contraction of muscle fibers. This part is a little bit confusing for me, but we'll struggle through it together. So an explanation of how skeletal muscle contracts is provided by the sliding filament model. And I have a feeling that she's probably not going to want us to know how to explain this to her, but just know the definition of that, because she really didn't hit hard on that, and I really don't feel like that's something she's super concerned about. But during to this model, during contraction, the thick and thin myofilaments in a muscle fiber first attract to one another by forming cross bridges that then act as levers to ratchet or pull myofilaments past each other. So if we look back at the myofilaments that we were talking about, the thin and the thick, myosin and actin in this picture back here, basically the sliding filament model is talking about how they're going to move together to make the muscle do what it needs to do. So if we bump down, the connecting bridges between the myofilaments form only if calcium is present. I don't have a mnemonic to remember that. You just have to know it. So in a relaxed state, calcium ions are stored within the smooth endoplasmic reticulum and in the muscle cell. And when nerve signals stimulate muscle fiber, ER releases calcium ions into the cytoplasm. So the calcium ions bind to the small blocking protein associated with the thin filaments and permit actin to interact with myosin. After the myosin heads connect to actin, they pull and release and then pull again. The ratcheting of myosin heads thus pulls the thin filaments towards the center of the sarcomere, producing the muscle contraction. So that's back on 153 in the way that these are working together to make your muscle move. So I don't think she's going to get too deep into that. I think she just wants you to know what the model is called to explain how this works. So the contraction process of a muscle cell also requires energy. The energy supplied by glucose and other nutrients The energy must be transferred to myosin head by the adenosine triphosphate or ATP molecules. So that's old stuff, and you will never stop hearing ATP. It's going to be continued forever. ATP is energy. ATP is energy. So we have to have ATP for these to work. Oxygen is required to efficiently transfer energy to ATP and make large amounts available to the myosin heads. So muscles have a high oxygen requirement. So if we are not getting a lot of oxygen, we're not breathing well, our muscles aren't going to want to work well either. So if you think about it, you have to be able to breathe to get oxygen, but you need those muscles to be able to breathe. So everything works together. Okay, so in addition to oxygen carried to muscle fibers by the hemoglobin in blood, muscle fibers contain myoglobin, that oxygen storing pigment similar to hemoglobin. During the rest, oxygen from the blood is stored in the myoglobin found in muscle fibers. The combined contributions of hemoglobin and myoglobin promote a maximum recharging of energy-containing ATP molecules. So this is referring us to chapter 17, but I didn't look at chapter 17. The only reason I knew what the heck they were talking about right here is because I did the outline for chapter 12, which is blood, and it confused the mess out of me because I hadn't heard of hemoglobin before. I don't know if we looked at that in another chapter, but I sure as heck did not. So the way that I remembered it is in your body, you have to have red blood cells to carry oxygen. That is their prime, that's their job. So if I have a blood vessel and I'm carrying blood, I'm going to have these red blood cells, and they're kind of biconcave shape, so the middle is going to be a little bit dipped in. The outer edges are going to be bigger, and these are erythrocytes or red blood cells. So red blood cells carry oxygen all through your bloodstream. But when you look at the blood chapter, it tells us that hemoglobin is what keeps the oxygen in the red blood cell and carries it through. So the red blood cell is just the car, it's the carrier. This is driving through our veins. Oxygen is the passenger. So this is who's riding in our car. And then I'm going to say that hemoglobin is the seatbelt. It's kind of holding us in place, holding the oxygen in place and helping to carry us through. So that's not a super, super big concept that you need to know, but if you're wanting to connect that, that's basically what that is. So in muscles, it's just a little bit different. And myoglobin works with the hemoglobin to carry oxygen through the muscles. So we're not in a vein anymore, we're in a muscle, if that makes any sense. So hemoglobin is just the seatbelt that's strapping in the oxygen on the red blood cell to move it through our veins and arteries. All right, so now we're going to jump over to functions of skeletal muscle. These are not very in-depth, and that's totally fine because I'm not wanting to stress about it anymore. So basically we have a few different types of functions for the skeletal system, and there was a couple people getting confused on these because it gives you one function and it didn't seem like it was very necessary. We'll get to that in just a minute, but the main is movement, posture, and I believe heat production. I need to look at the next page. Yes. Okay, so the other option at the end of this was fatigue. And I was like, well, that's not doing me any favors, but that's just letting you know a function of the skeletal muscle. I'm sorry. Okay, so by producing continuous tension on the skeleton, muscle tone is a system that also helps maintain a stable body position or posture. It also helps us produce heat and maintain homeostatic balance of body temperature. So I'll kind of just bump through this. I'm not going to read that straight down because it's not going to be like that on the test. So basically when it's talking about muscle tone, I think of when we are doing checkoffs and the teacher's asking us to pull the bed up to our working height because we don't want to stress our muscles. So muscle tone is going to be relating to posture. We want to sit up straight. We want to work our muscles the best way that we can. It's the most beneficial to us. It's not putting a strain on them either, if that makes any sense. So when you come into a patient's room, you're going to want to lift the bed up so you're not straining your muscles. You want to practice good posture. Pretty simple concept. So movement, obviously, muscles move bones by pulling on them Because of the length of skeletal muscle can shorten as its fibers contract, the bones to which the muscle attracts to move closer together. So if you're thinking about your arm, when you are contracting a muscle, it's going to shorten, and it's going to move your bones closer together. So these bones are now closer together. So there's a simple rule to remember, and that's a muscle's insertion bone moves towards its origin bone. So if I am doing the diagram like I did with my leg, we'll use our arm this time. This is going to be the origin bone. This is going to be the insertion bone. Why? Because this is more stationary to my body. It's more stationed and stable to where it's at. So then I have my my joint, and then I have my insertion bone and these muscles that connect it. So when I flex my muscle, when I contract my muscle, it brings my insertion bone closer to my origin bone. It's going to decrease the angle, which is another thing that we talk about in like dorsiflexion and stuff here in just a little bit. So muscles can also produce tension as they extend. When muscles lengthen under tension for a muscle insertion, as the muscle insertion is pulled by a load away from the origin. So it says, for example, when you lower a heavy bowling ball from your shoulder, the muscles in your arm produce tension as they lengthen and allow you to gently lower the ball rather than just completely dropping it down. So tension during muscle lengthening is often called eccentric contraction. And the way that I thought about that was eccentric kind of sounds like a wild word to me. So it's pretty eccentric that we can lower a bowling ball without dropping it to the floor. I don't know. It's probably dumb, but that's just how I remembered it. So voluntary muscle movement is normally smooth and free of jerks and tremors because skeletal muscles work together in a coordinated team rather than individually. So this is what we were saying earlier. Everything's trying to work together. It's not trying to get crazy. You want to be able to perform a function normally without being stopped. I'm going to take just a second to check on the group. Okay. So all of the muscles contracting simultaneously, of all of them, mainly one is responsible for producing a particular movement, and this is the prime mover. So we're still in movement, and we're going to go over to prime. So when you think prime, I think Amazon Prime. My husband pays for Amazon Prime. I think it's too much, but that's just what he does. Prime as in he's probably getting like the main deals. He's getting to watch all the good shows because he's paying for them. He's at the top of it. Of Amazon's list with Amazon Prime. So this is going to be like the main dude up here. So you have the prime mover for that movement, and then the other muscles that help to produce the movement are called synergist muscles. So we'll just put that second. And then you can also say synergist second. So prime is going to be top dude, and then synergist is going to come second, and they're just going to be helpers. So prime movers and synergist muscles at a joint contract. Other muscles called antagonist muscles relax. So when antagonist muscles contract, they produce a movement opposite to that of the prime movers and the prime movers synergist muscles. They don't really talk anymore about that. Antagonists kind of confused me because there wasn't enough on it, but just know that there's prime and then synergist. These two are going to work together as a team. And then you have antagonist, and this just kind of sits in the background, and these are going to relax. So we'll just say relax over there. Okay. Let's see. Okay, it says on figure 7 or 8-7, locate biceps brachii, brachialis, and triceps brachii muscles. These are involved in bending and straightening the forearm and the elbow joint. So this, I'm not going to get super deep in. It's going to talk about it later. It's basically letting you know that there's a certain amount of muscles and a certain amount that's needed to be able to do different small functions, and those are those muscles. So now we're going to go down to posture. Low strength muscle contraction is called muscle tone or tonic contraction. And so this is going back to the first little paragraph that we saw under functions of skeletal muscle. Also, I'm sorry, I haven't been keeping up, but we're at the bottom of page 155. So because relatively few of the muscle fibers shorten at one time in a tonic contraction, the muscle as a whole does not shorten and no movement occurs. Consequently, tonic contractions do not move any body parts. They do hold muscles in position. However, in other words, muscle tones maintain posture. So even if we're not walking, we're still using our muscles. They're just kind of sitting in the background and keeping us sitting upright and sitting normal, and that's posture. So we still use muscles even though we're not moving, but that's what it's saying when they're not contracting. So is that what the word was? Tonic. Yeah, tonic contraction. I don't know of a way to remember that, but it's a small word, and I don't want to say it's going to be insignificant, but just know the definition of it. So tonic is just posture. Tonic contraction is posture. We're just sitting and we're not using that muscle. Good posture means that body parts are held in the positions that favor best function, balance and distribution of weight, and therefore put the least strain on muscle, tendons, ligaments, and bones. So you could even think of it as sleeping. Tonic, I'm getting ready for tomorrow and I'm resting. My muscles are still being used, they're just not being contracted. Let's see. Skeletal muscle tone maintains posture by counteracting the pull of gravity. Gravity tends to pull the head and trunk down and forward. Obviously, as I'm sitting here, I'm not using good posture. It pulls your back and neck muscles just hard enough in the opposite direction to overcome the force of gravity and hold the head and trunk erect. So we're going to go to heat production. So this tells us that fever or elevation of body temperature of only 1 or 2 degrees above 98.6 is almost always a sign of illness. So you can look at someone and tell if they have a fever. A lot of times you can feel someone's forehead. And it's not necessarily a bad thing. It's just letting us know that our body's having to work a little bit differently, kind of swap up gears so that it can fight whatever's going on. So it says decrease below normal. Any decrease below normal is a condition called hypothermia. And so I know we've heard this a couple of times, especially if you watch Grey's Anatomy. That's my number one. Hypothermia is going to be a contraction of muscle fibers that produce the heat required to maintain body temperature. So if you are, say you fall into a lake of ice that's frozen over, hypothermia is pretty serious because your muscles are giving out all of the heat that they can to stay warm. They're working overtime. They're acting hyper. So energy required to produce a muscle contraction is obtained from ATP energy. Some of the energy transferred to ATP and release during muscular contraction is used to contract the muscle fibers. However, much of the energy is lost as heat during its transfer to ATP, and this helps us to maintain our body temperature at homeostasis. So basically, we get too cold, our body's going to start shivering, and it's going to start working those muscles. And I mean, you can feel it too. If you sit here and do this with your hand, you feel heat production. That's basically what your muscles are doing inside. When you start shivering, your muscles are working together, and that's to produce heat. It's just a natural thing that we do when we get cold. So now we're going to jump down to fatigue. This was another pop quiz question that I had on the website. If muscle fibers are stimulated repeatedly without adequate periods of rest, the strength of muscle contraction decreases, resulting in fatigue. So during exercise, a small amount of stored ATP or energy is required for muscle contraction, and it's quickly depleted. So if you think about going to the gym and you're really kicking it, working really hard, all that energy is going to go very, very fast, and it's just going to leave. So the formation of more ATP results in the rapid consumption of oxygen and nutrients, often outstripping the ability of the muscle's blood supply to replenish them. So you're using this ATP very fast. You're getting rid of a lot of oxygen because you're working really hard. So what does your body have to do? Your body has to find a way to get more oxygen. So this is where we come in with oxygen debt. So if I'm working out, I'm wearing myself out, all that energy is leaving my body, I need more oxygen. I'm going to start breathing more. I'm going to start trying to get as much air as I can into my lungs so that can get into my bloodstream so that I can start breathing better, which also gives more energy to my muscle fibers. I want to be able to breathe. I want to be able to keep working out. So oxygen debt is just telling your body it's owed. Your body's basically saying you owe me some more oxygen because you've been making us work and it's all gone. So it continually increases metabolism that must occur in a cell to remove the excess lactic acid that accumulates during prolonged exercise. So labored breathing after a succession of exercises is required to pay the debt of oxygen required for metabolic effort. And then it says psychologists or physiologists, I'm sorry, use oxygen debt with a more technical name, and this is excess post-exercise oxygen consumption. I guess they're super extra. Basically, the reason why we start breathing heavily is because we've lost oxygen. We're trying to gain that back. And the reason why our muscles get super sore is because once you get to a point of using all of the ATP that you have and you're still trying to get that oxygen to your body, the next thing that's going to kick in is the lactic acid. And that's what causes the burning feeling when you get done working out and you really regret going. That's just it's a side effect of this. So I'm going to go down to the role of other body systems in movement. Is there anything that you guys want to go over before I move on? I don't know if I'm moving too fast or what, but it kind of gets a little bit easier throughout here. It kind of seems to start with the harder stuff and then kind of levels out, but I just want to make sure that I'm covering everything. Everybody good? All right. So we'll keep going. We're at the bottom of 156. So remember that muscles do not function alone. Other structures such as bones and joints must function along with them. Most skeletal muscles cause movements by pulling on bones across movable joints. So that was the picture that we drew with the two bones, the joint in the middle, and then the way that the muscle overlaps, kind of like a rainbow. However, the respiratory, circulatory, nervous, and muscular and skeletal systems all play essential roles in producing normal movements. So a person might have perfectly normal muscles and not be able to move normally. So they might have a nervous system disorder that shuts off impulses to certain skeletal muscles, and this results in paralysis. So MS or multiple sclerosis would be an example of this. And then other conditions such as a brain hemorrhage, brain tumor, or spinal cord injury. I would definitely know that little bit because she may pull that. Paralysis is you have muscles and they're normal muscles. Everything's fine with those, but there could be, say you got into a car accident or some kind of accident and it severed a nerve. Your muscles still look fine. They still give the aesthetic part of it, but they're not going to connect to parts of your brain that are going to help them move. They're not going to be able to perform their function. So this would be paralysis. So skeletal system disorders, especially arthritis, have a disabling effect on body movements. Muscle function then depends on the functioning of many other body parts. So I'm going to go ahead and jump ahead. It's not in this chapter, it's in the nervous system, but there's a little blue box where it talks about somebody with Parkinson's disease, and this is where that would tie in. I'd like to know what page it's on. There we go. On page 187, where it's, there's a research issues and trends blue box. In Parkinson's disease, your muscles are totally fine. There's nothing wrong with your muscles. It's just a disconnect between your muscles and your brain. And so it's going to cause them to be shaky and uncontrolled. And this was also something that we had talked about before in our posture. Posture is what keeps our muscles normal at a resting state. Well, in someone with Parkinson's disease, this can't happen. There's not a connect where it needs to be. And so these muscles aren't going to perform the functions that they need to. Okay, so I'm going to go down to motor unit. This stuff kind of starts to run together, so we'll suffer through that together. Some of these words kind of start to get a little mixed up, but before skeletal muscle can contract and move a bone, the muscle first must be stimulated by a nerve impulse. Muscle fibers are stimulated by a nerve cell called a motor neuron. Any single motor neuron can stimulate more than one muscle fiber. However, each muscle fiber communicates with only one motor neuron. So the point of contact between the nerve ending and the muscle fiber is called a neuromuscular junction. And keep in mind that motor neuron and muscle fiber membranes do not actually touch. There's a fluid matrix between them. So this is in figure 8-5. The muscle fibers are wrapped in this sheath, and there's going to be a junction between the motor neuron and the myelin sheath. I'm really trying to explain this the best way that I can. Okay, so figure -5 is the best way that I know how to just refer you to it because it's not a large part that needs to be known. this is basically just giving you the gap. So let's see if I can draw this. Bear with me, y'all. I'm trying. All right, so motor unit. Let's see. All right, so you got your tube. This is inside of a muscle. And then you've got all these little pieces coming out of it. And the little pieces that are coming out are your muscle fibers. So these are what are working together to pull your muscles. And then you have your motor neuron. So this is going to look like a lot of the stuff that you're seeing in the nervous system. But these motor neurons are connected. I say they look kind of like tapeworms. They gross me out. I don't like looking at them, but that's just me. And then it's got these little finger projections that kind of come off of it, and at the bottom of them, they kind of look like teardrops to me. But those are going to connect. And this, okay, so I'm going to go back down to this paragraph. The point of contact between nerve endings and the muscle fiber is called the neuromuscular junction. So these little pieces are neuromuscular junction. Okay, so motor neuron and muscle fiber membranes do not actually touch. There's a fluid matrix between them. So see how there's a gap right here? This is going to be a fluid matrix between these. They're not actually touching one another. So these little pieces right here will say. And if you remember in the past chapters, the fluid matrix is kind of just like a little buffer area. I kind of associate it with the synovial fluid or bursae. It's just lubricating. Okay, so let's see. Okay, so before skeletal muscle can contract, before this can move, and this is in a bigger piece back here. This is just a part of this pulled out, and then that's pulled out of this. Muscle fibers are stimulated by a nerve cell called a motor neuron. Okay, so for this to stimulate this, oh no, I'm sorry. For this muscle to move, this has to stimulate this. So we have the gap between the nerve endings or the neuromuscular junction, and then we have the fluid matrix between them. The signal chemicals called neurotransmitters are released by the motor neuron in response to nerve impulse. So then here we're going to have little neurotransmitters. That's how they're going to, because they're not touching each other, they can't connect, they're touching each other. They're going to send out neurotransmitters, kind of like little mailmen, to communicate. So the type of neurotransmitter operating in each NMJ is called acetylcholine, or ACH, and it's released. It moves across the NMJ and triggers events within the muscle fiber, resulting in its contraction. And a single motor neuron within a muscle fiber, it innervates, is called a motor unit. So this is a motor neuron. This little piece is a motor unit. Okay, so all we're trying to do is move this muscle. For this to move, this has to stimulate this. Is that making sense? Is this making sense to everybody? I'm trying my best, I promise. All right. So in our bodies, we have both small and large motor units. The small motor units, a given motor neuron control only. In small motor units, a given motor neuron controls only a few muscle fibers. Small motor units are common in the eye and muscles and offer precise control. In large motor units, a given motor neuron controls hundreds or maybe thousands of muscle fibers. So large motor units are common in the thigh and are activated in high intensity activities. So that's not going to be hard to remember. The small ones are going to be teeny parts like your eyeball, and the larger ones are going to be parts like your thigh that you're trying to move so you can walk into the grocery store. Okay, so muscle stimulants, let's see. In a laboratory setting, a single muscle fiber can be isolated and subjected to stimuli of varying intensity so that it can be studied. Each experiment shows how a muscle fiber does not contract until its applied stimulus reaches a certain level of intensity. So when we're working with this situation right here, it has to reach a minimum level of action for this to contract. So for the muscle to contract, we have to reach what, y'all see this down here. We have to reach this threshold stimulus. And I hate that name because it doesn't feel like it goes with this at all, but it is what it is. So we have to be able to reach that for this muscle to contract. There's a lot of steps that have to be taken before we can even stand up and walk. And it's crazy because we don't even get to control them. It just happens. So when a muscle fiber is subjected to a threshold stimulus, it contracts completely. On the other hand, if a threshold stimulus is not reached, the muscle will not contract. So they're all or none. There can't just be a little bit. It can't just be a lot. Like it's going to be all or nothing. So muscles composed of many muscle fibers that are controlled by different motor units and have different threshold stimulus levels. So although each muscle fiber in a muscle, such as biceps brachii, responds when subjected to a threshold stimulus, the muscle as a whole does not. So it says that this fact is tremendous in importance in everyday life. It allows you to pick up a 2-liter bottle of soda or a 20-kilogram weight because different numbers of motor units can be activated for different loads. So basically, how big this is going to depend on whether I pick this pen up or whether I'm picking my book up. So now we're going to go down to types of skeletal muscle contractions. Johnny, you leave this up. Does anybody want this up or can I erase it? All right, I'm going to go ahead and erase this because you guys can come back and look at it later if you want to. All right. So we're going to go to the types of skeletal muscle contractions. So we're looking at types. We have twitch. We have tetanic. We have isotonic and isometric. If you don't mind, I'm going to get my notes because I wrote notes for this. I would like to look at them. Okay. Yes. Okay. So I just condensed this out of this, the paragraph that it gave us because there's a couple paragraphs on it. But basically, okay, twitch is going to be exactly what it says. It's going to be jerky. So if you have me, when I'm drinking all these dang energy drinks so I can stay away from nursing school, I'm going to be twitchy, so it's kind of jerky. All right, so tetanic is going to be a steady or sustained, and this is tetanus. And this one's going to be steady. Okay, isotonic is movement in the joint. Muscle changes length, and insertion is relative to origin. So when we think isotonic, we're moving. So this is basically, if you break down what that just said, it is walking. It is picking up something. So isotonic, the movement in a joint, muscle changes length, and insertion is relative to origin. So whether we're moving it out or we're moving it in, these two are correlating with one another, and we're working that joint. So this one is movement in joint, muscle changes, and insertion and origin. We'll say that. All right, so then isotonic is broken down into two other groups. So this gives us a better idea of that definition. So if this is related to moving things, are you making something go farther away or are you making something closer? So this is going to be broken down into concentric and eccentric. Okay, so concentric is going to be when a muscle shortens, and then eccentric is going to be when a muscle lengthens. So we're going to say short and long. Okay, so the way that I would remember this would be concentric is going to be like contracting. So here's my arm. Concentric, isotonic would be moving this, and you're shortening these muscles. They're shortening out. They're coming closer to you. They're constricting. This is concentric. Eccentric would be extending your arm to someone. This is lengthening a muscle, but it's also still an isotonic movement. So that's how I would remember that. All right, and then we'll go up here for isometric. She didn't really want us to hit super, super hard on these, but in case she throws one on there, we'll do it. All right, so muscle contraction with no movement. And the way that I look at that was the example in the book. It stuck with me. If you come up to a wall and you push on this wall, none of your muscles are contracting, but you're still putting off a force. Or I'm trying to think what the word they used was. Let's see. Equal measure. Okay, so there's tension. There's going to be tension. You're going to feel that pressure in your arms, but you're not moving any kind of muscle in a contraction at all. So there's going to be tension, but no movement. And this would be pushing a wall. Okay. All right. I'm finished with that. Okay, so like we said, isotonic is going to be probably the one that we use the most. This one's going to be walking. I'm going to put sports. Any kind of high movement like that is going to be the isotonic because you're going to be doing a lot of movement. So equal measure is going to be for isometric. Equal measure. Figured out my handwriting. Let's see. Okay. So it says, although the muscles do not shorten, we're talking about isometric right now, tension within them increases. And because of this, repeated isometric contractions make muscles grow larger and stronger up to a point. So pushing a wall or a movable object is a good example of this. Although no movement occurs, it doesn't shorten, but there's tension inside of it. And so that's when we transition down into the effects of exercise on skeletal muscle. If it's okay with you guys, do you want to take like a 5-minute break? Go get something to eat, go get something to drink, and come back. Let's see, it's 4:11 right now. Y'all want to come back like 4:15, 4:16? Is that cool with y'all? Okay, good deal. All right, I'm gonna run and go get something that's not an energy drink, and I'll be right back. All right. Just comment when you're back and ready to stop. All right. Is it okay if I go ahead and get started? All right. Good deal. All right, so now we're moving down to effects of exercise on skeletal muscle. These are kind of self-explanatory. So benefits of regular, properly practiced exercise include improved muscle tone, better posture, more efficient heart and lung function, less fatigue, and looking and feeling better. So we've probably heard this our entire life. Exercise is good for you. Exercise is good for you. Eating right, exercise is good for you. Which is another reason why you can't just diet and just eat good. You have to be able to stress your muscles and work with those, because they're not just for looks. It has to do with your heat production, has to do with your posture, has to do with pretty much the longevity of what you were born with. During prolonged inactivity, muscles usually shrink in mass in a condition called disuse atrophy. And so atrophy, you're going to hear a lot and other things too. But when I think of this, I think of when I worked at the nursing home. There was a lot of patients who were kind of, I don't want to say bedbound, but they couldn't get up like they used to. And so they weren't able to use those muscles. This is another reason why we like to do, trying to think of a word for it. We work out their muscles where we come in and assist them in stretching so that they don't get contractures. And contractures is when you kind of just stay in one spot and you don't move around as much. But disuse atrophy is going to be when your muscles aren't being used, they're not being worked, and they're going to kind of start to drift out. So in patients who have who are paraplegic or quadriplegic, a lot of the times, if you see somebody that's paralyzed from the waist down or they're in a wheelchair, their legs are going to be a lot tinier than the upper half of their body because they're not using these muscles and they're atrophying. They're giving out. They're going away. So on the other hand, the opposite end of that spectrum is hypertrophy, and this is an increase of muscle size. So when I think of hypertrophy, I think of The Rock. He's a favorite. So he has definitely been working in strength training. That's the next bolded term. So strength training would be, the whole point is for the aesthetic of it and to be able to use the bulk of your muscle. So hypertrophy, bigger muscles. So this is just working out because you want to look buff. All right, so muscle hypertrophy can be enhanced by strength training, which involves contracting muscles against heavy resistance. So this goes back to where we were talking about an isometric contraction. So a push-up, you're pushing down on the floor to push yourself back up, and this is going to cause hypertrophy. So this is going to make your muscles bigger the more that you're doing that. So you're pushing on something that is heavy in resistance. That floor is not going to come towards you. So isometric exercises and weightlifting are most common strength training activities. This type of training results in increased number of myofilaments in each muscle fiber. Although the number of muscle fibers stays the same, the increased number of myofilaments greatly increases the mass of the muscle, so that's why they look so bulky. Endurance training, or often called aerobic training, does not usually result in muscle hypertrophy, but it increases the muscle ability to sustain moderate exercise over a long period of time. So when I think of this, I think of the people that run in my neighborhood. God bless them, because I can never do that. Every morning, I'm getting ready to get my car and leave, and there's this man that runs around, and he is so tiny. He's just a tiny dude. He has to be like at least 2 feet taller than me, but he's so thin, and I'm thinking, how is it that he is running? His leg muscles should be freaking huge, but he's not running and putting a lot of the running is not causing the resistance for him to have hypertrophy. This is not for a purpose of him having larger muscles, but I promise you, he could climb the stairs in my house without having shortness of breath, and I cannot do that because he has had endurance training. He can endure a lot more, or aerobic training is what it's called. So endurance, you're going to be able to withstand a lot more because you have worked your body to this point where it can handle a lot more, and you're not worried about the muscle part of it. And aerobic training is another word for that. So when I think of aerobics, I think of water aerobics, all the little old ladies with the caps on their head and they're in the big swimming pool and doing all this fancy stuff together. That's what I think of. So they're not doing anything to gain big muscle mass. They are just doing that so that they can kind of keep their body within use and stuff. It's usually elderly people that you see doing that. And that's just so that they can endure ADLs. So let's see. Running, bicycling, or other primarily isotonic movements increase the number of blood vessels in a muscle without significantly increasing its size. So with this, the difference between this and strength training is going to increase the number of myofilaments. Aerobic training is going to increase the number of blood vessels. Increased blood flow allows more efficient delivery of oxygen and glucose to muscle fibers during exercise. Give me just 1 second. Let's see. All right, so Callie can't get on here, but she's reminding me of some things to bring up, especially when we get to the cranial chapters. And if there's anything that you guys need to ask her, she's very much available for you to message her as well, especially if there's something that I'm not explaining very well. You're not going to hurt my feelings at all if you go to somebody else and ask for help at all. You learn how you learn, and that's totally fine. So the increased blood flow allows more efficient delivery of oxygen and glucose to muscle fibers during exercise. And aerobic training causes an increase of number of mitochondria in the muscle fibers. This allows production of more ATP, which is needed to fuel prolonged activity. So basically what this is saying is the little red blood cell car that we drew with the oxygen, that's me and you sitting on the red blood cell. And then the, I'm sorry, I'm just lost my place. The word that I'm looking for is hemoglobin. That's going to help carry the oxygen. Glucose is a sugar. This gives us energy. So you think of eating, fruits, they have sugar in them. This is going to give you energy. Well, this gives energy to the muscle fibers during exercise. It causes an increase in number of mitochondria. And as we know from the first or second chapter, mitochondria is the powerhouse of the cell. So this is what's giving the energy. This is kind of like a generator, if you will. It's going to keep it going. So you need a lot of ATP to have oxygen to be able to withstand these things. So these people that are doing the aerobic training probably have a lot better lung capacity than we do. One more. Okay. All right. So now we're going to run over to movements produced by skeletal muscle contraction. So a particular type of movement that occurs at any joint depends on the muscles acting at that joint in their origin and insertion points on the shapes of bones involved in the joint type. Muscles acting on some joints produce movement in several directions, whereas some acting on other joints produce only limited movement. Meaning, so the muscle and the joint for my shoulder, I can move it in lots of different areas. It gives me a lot to work with. Whereas in my knee, it may not be as good. It's going to be more of like a stationary kind of position. Okay, so we're going to go over to angular movements. This one we can kind of blow through because we have already gone over this. So flexion is a movement that decreases in angle, and the way that I think of it is flex your muscle. flex your muscle, you just decrease that angle. It's a tiny little angle, all right? And then extension is going to be the opposite. You just extended your hand to someone and you have opened this part of your arm, all right? So now we're on page 160, and there's examples of this. All right, so abduction is moving a part away from the body, from the midline of your body. So if I'm standing and I move my arm away, somebody has abducted my arm, they've taken my arm away from my body. So this is moving it away from the midline of my body. Adduction is moving the arm towards the body. So I've added an arm, adduction, I've added something towards the midline of my body. Circular movements are going to be rotation. It's a movement around a longitudinal axis, and you can rotate your head and neck by moving your skull from side to side. So this is rotation. Circumduction is a part that moves so that its distal end moves in a circle. So when a pitcher winds up a throw ball, she circumducts her arm. So this is circumduction. This is moving. All right, so supination and pronation refer to hand position. So we had supine, where we are laying down, up. And then we had prone, where we were laying down. So we're going to use the same for our hands. So supination is going to be when my hand is facing the front in an anatomical position. And then we have pronation, where it's facing the back in an anatomical position. All right, so then some body parts, such as the foot, are difficult to describe in ordinary terms. But dorsiflexion and plantar flexion, we use these during our physical assessment. So dorsiflexion, if this is my foot, dorsiflexion is going to be moving the foot towards you. So if this is the bottom half of my leg, this is my kneecap. Dorsiflexion is going to be moving towards you. Your toes are going to be lifting up. Plantar flexion is when you're planting your foot down. You're planting your toes down into the floor. That's plantar flexion. All right, and then we have inversion and eversion. So inversion turns the ankle so that the bottom of the foot faces towards the midline. So if these are my feet, inversion is going to move them towards the midline of my body. Eversion is going to push them out. So when you stand on your feet, and you can move them in and out. Eversion or inversion at eversion. So in moving in, eversion is out. All right, skeletal muscle groups. These I don't have a way to help you memorize. This is definitely know the bolded ones. I'm not going to say that you have to memorize everything in here, but you are going to need to know the bolded terms. She's going to throw some of these in here, I know. Then I want to go ahead and say, first and foremost, please make sure that you write down the ones for IM injections. These are the ones she specifically said. We're definitely going to need to know these. And this is the deltoid, which is going to be out here. Vastus lateralis, which is going to be in your thigh. And then ventral gluteal, which is going to be, I believe it's on the back, on your posterior side. I did not circle that one in there, but I do have it written down. So we'll start at muscles of the head and neck. Muscles of facial expression allow us to communicate many different emotions non-verbally. So you have a frontal muscle, which allows you to raise your eyebrows and surprise and furrow the skin on your forehead to a frown. The orbicularis oris is the kissing muscle, so it's going to pucker your lips. Zygomaticus elevates the corners of the mouth and lips, and this is the smiling muscle. So these muscles are going to pull. Muscles of mastication are responsible for closing the mouth and producing chewing movement. So mastication refers to chewing. So mastication, chewing. Two largest muscles are going to be your masseter, and it elevates the mandible. And the temporal, which is the masseter and closing the jaw. So masseter is, let's see if I can find a picture of it. Okay, masseter is going to be right here. And then your mandible is down here. And these are going to work together so you can produce mastication, which is chewing. All right, so sternocleidomastoid and trapezius muscles are in figure 8-10 and 8-11. Two sternocleidomastoid muscles are located in the anterior surface of the neck, and these are right here. You can see these muscles like this. They originate in the sternum, and then they pass up and across the neck. In the insertion on the mastoid process of the skull, working together, they reflex the head to the chest. Excuse my double chin. So these muscles are going to help to pull those. If one contracts, the head is both flexed and tilted to the opposite side. So these muscles are still controlling this. The triangular shaped trapezius muscles in the line from each shoulder to the neck on its posterior surface. They have a wide origin extending from the base of the skull down the spinal column to the last thoracic vertebra, where contracted, they move, they elevate the shoulders and extend the head backwards. So these muscles can do this. And by the way, y'all, I went to the chiropractor today and I'm feeling great. I'm feeling myself because I haven't been fixed, and he fixed all these muscles for me, and I'm ready for tomorrow. So we're going to move over to muscles that move the upper extremities. These dudes right here. Each upper extremity is attached to the thorax by the pectoralis major. When you think pecs, think of like a surfer dude. He's got pecs. These are the pectoralis major muscles. They cover the upper chest and the latissimus dorsi muscle, which originates from structures in the lower back. So latissimus dorsi are going to be the muscles on your back, kind of towards your side. All right, and then we see figure 8-2. I'm not going to say know all these, but I am going to say peek over it and then know all the ones in here that are bolded the best that you can. Not going to have to memorize though, but at least maybe try and figure out where they are on the body. And some of the names will tell you. So deltoid muscle forms a thick, rounded prominence over the shoulder of the arm. That was a question that Miss Hutt gave us on her little test that she gave us today. So deltoid is going to be right here, and this is where we do IM injections. It's kind of like a little thick area. So this is where we do IM injections. It's a powerful abductor of the arm, so you're going to have these bad boys to lift this up. So biceps brachii is a two-headed muscle that serves as the primary flexor of the forearm. So biceps brachii is going to be the fat part right here. This is going to be what you see a lot of on the bodybuilders. And so this serves as a primary flexor of the forearm and originates from the bones on the shoulder girdle and inserts in the radius of the forearm, which I don't think we're going to have to know all that. So triceps brachii is in the posterior surface. You got biceps and triceps. So B's come first in the alphabet. B is in the front. T comes later, and so that's in the back. Posterior to back surface of the arm. It has three heads that have their origins on the shoulder girdle and insertions on the ulnar-cranial process of the ulna. You know all that. Muscles of the trunk. So muscles of the anterior or front side of the abdomen are arranged in 3 layers. I would probably know these. So you have a rectus abdominis, external oblique, and aponeurosis of external oblique. Or, let's see. Yeah. Okay, so on page 166, it says 3 layers of muscle in the anterolateral side. Abdominal walls are arranged as follows. Outer is external, middle is internal, and then outermost is transversus, or innermost is transversus, I'm sorry. So band or strap-shaped rectus abdominis muscles run down the midline of the abdomen through the thorax and to the pubis. And this is figure 8-12. So in addition to protecting the abdominal viscera, the rectus abdominis flexes the vertebral column. So these are your respiratory muscles. So respiratory muscles are going to be discussed later in another chapter, but intercostal muscles we have already gone over, and those are the muscles between your rib cages. So if you put your hands on your rib cage, take a deep breath, you can feel your ribs moving, and those muscles between your ribs are what's helping to move that. And these are intercostal, so in between muscles, in between your rib cage. So then you have a diaphragm separating your thoracic and abdominal cavities. And so the way that I remember diaphragm is the bottom of your bra is where your diaphragm would be. And this is what helps to change the shape and size of the chest during breathing. So as a result, air is moved into and out of the lungs. So you have to have your diaphragm to be able to breathe completely, get a full breath, and to exhale. So muscles that move the lower extremities are the iliopsoas. These are deep within the pelvis and lower vertebrae to insert lesser trochanter of the femur. Generally classified as flexor of the thigh, an important postural muscle that stabilizes the trunk and keeps it from falling backwards when you stand. So the thigh is fixed so that it cannot move, and the iliopsoas flexes the trunk. So this is an example when we're doing sit-ups. Gluteus maximus is the outer contour, much of the surface of the buttocks. Adductor muscles originate in the bony pelvis and insert the femur. They're located on the intermediate sides of the thighs, and these adduct the thighs or bring them together. 3 hamstring muscles. I would know these because I feel like this came up in a lot of quick checks, the question at the end of the chapter and for the workbook. So I would definitely know these. There's a semimembranous, semitendinous, and biceps femoris. So when I think biceps femoris, it's around your femur. Acting together, and they serve powerful flexors in the leg. So quadriceps femoris is a muscle group that covers the upper thigh when you think quads. 4 thigh muscles, the rectus femoris, and 3 vastus muscles extend the leg. 1 component of the quadricep groups has its origin on the pelvis, and remaining 3 originate to the femur. All 4 insert to the tibia. The vastus intermedius is covered by the rectus femoris and is not visible. Again, I don't think we're going to know all of that. I think she just wants us to know basically where these are. And I don't think we're going to be labeling, but she's going to want us to know, because when it comes to intramuscular injections, we're going to have to make sure that we know what we're talking about, so we don't stick a needle in somewhere where it doesn't belong. So tibialis anterior muscle is located on the anterior or front surface of the leg. It dorsiflexes the foot. Gastrocnemius is the primary calf muscle. So people that have really big calves have big gastrocnemius muscles. It's 2 fleshy components arising from both sides of the femur. It inserts through the calcaneal or Achilles tendon in the heel bone or calcaneus, and the gastrocnemius, this is hard, y'all, is responsible for plantar flexion of the foot. So since it's used to stand on a tiptoe, it's sometimes called the dancer's muscle. That might be some pretty cool information that we would need to know. The 3 groups of muscle are called fibularis group or peroneus group, and are found along the sides of the leg. They evert the plantar flex in the foot. Long tendon that forms from 1 component of the group is the fibularis longus muscle tendon, and it forms support arch for the foot. So that's the end of this chapter, but I kind of want to go back through real quick and grab some things from these boxes in here, like the research issues and trends and stuff, so that we make sure we covered everything. Let's see. All right, there's a green box on 155. Let's see. Nutritionists use information to advise athletes and others on what to eat when to maximize muscle strength and endurance. So nutritionists will think of that. They have a lot to do with muscle function. Physicians, nurses, physical therapists, and occupational therapists apply information when they're helping clients with muscular problems such as myasthenia gravis and muscular dystrophy, to improve mobility and quality of life. So many other professions such as massage therapy, ergonomics, physical education, fitness, dance, art, and biochemical engineering. So any of these could have to do with this, and this might come later in our medical terminology book. We're going to need to know who does what. So then I'm going to bump over to 156, and there's a health and well-being, slow and fast and muscle fibers. She didn't really hit this, but slow fibers are red fibers because they are high content oxygen storing myoglobin. Fast fibers are white fibers because they're relatively little myoglobin. Intermediate fibers have characteristics between extremes of slow and fast fibers. So maybe just look at that. Page 159, there's a blue research issues and trends, enhancing muscle strength. Most obvious way of increasing skeletal muscle strength is by strength training, and it's regularly pulling against a heavy resistance. So this is basically going back over hypertrophy. Let's see. Okay, chapter 8, 162. This was a question in the workbook, I believe. Yes, it was a workbook, and it was talking about somebody who had a problem with her wrists. So this says, one common problem caused by repetitive motion is tenosynovitis, inflammation of the tendon sheath. Tenosynovitis can be painful, and the swelling associated with this condition can limit movement and affect parts of a body. For example, swelling of the tendon sheath around the tendons in the area of the wrist is known as carpal tunnel and can limit movement, or carpal tunnel syndrome. So when I think of this is a lot of people that have, it's 2020, y'all. Everybody's got desk jobs. We're all trying to work from home, and especially in this time with this virus. People that work on computers a lot are working the muscles in their wrists a lot because they're doing this all day. And so this area starts to get inflamed. The sheath around the tendon starts to get inflamed, and this is carpal tunnel. So carpal bones, or your wrist bones, tunnel. This is inflammation of those bones. So the next one is on 166, is a clinical application for muscle injury. So injury to skeletal muscles resulting from overexertion or trauma usually causes muscle strain. Muscle strains are characterized by muscle pain or myalgia and involve myalgia. and involve the overstretching or tearing of muscle fibers. And if an injury occurs in the area of a joint and a ligament is damaged, the injury may be called a sprain. Cramps are also in here, bruise and contusion. So I would look at those as well. All right, does anybody have any questions for chapter 8 before we take a little break for chapter 9? Also, y'all, it feels like 10 o'clock, and I'm in a room where all the curtains are closed, and it feels like it's time for me to go to bed, but it's literally 4:40. Excuse me. Does anybody have any questions from chapter 8? All right, I'm going to say let's take a break and start back at, we'll do 4:50. It's 4:42 now, so I'm going to take a break and come back at 4:50, and we'll start on the nervous system and try and bump through this as quick as we can. All right, so we'll go ahead and start back. All right, so we're going to start in the nervous system chapter. Y'all bear with me because this one is probably the most stressful out of all of them, I feel. But we're going to get through it. We just have to remember what she said and not get too far in depth with it because then we're going to stress ourselves out. And I don't feel like she feels making like that stressful of a test, so I don't think that it's going to be as crazy as we're feeling. So I don't know if anybody's printed off the PowerPoints or if anybody's printed off anything from the website, but that's how I've done literally this whole entire chapter is drawing things out so that I can remember it. And I'm going to do my best to explain it. Basically, all right, so this starts on page 175. It tells us that nerve impulses and hormones communicate information to body structures, increasing or decreasing their activities as needed for healthy survival. So we're going to bump down. There's two principal divisions. Let's go ahead and do this. Oh, I'm just rolling all over the place in this chair. All right, nervous system. We have CNS, and then we have PNS. So peripheral nervous system and central nervous system. And central is going to contain the brain and spinal cord. Brain, and then the cord down our back. All right, and then the peripheral nervous system is going to contain spinal nerves and cranial nerves. Okay. Let me bump back just a little bit. And I'm kind of going to jump around just a little bit, but I'll try and keep you up on where what page I'm on. So the PNS also has an autonomic nervous system. So we'll go down to this. And then this is going to branch off into sympathetic and parasympathetic. So we have parasympathetic. All right, so parasympathetic is going to be our resting state. We are at peace. This was a print-off that I did, and I picked up a lot of this from Miss Putt. But so parasympathetic, you're going to be at peace. Sympathetic, you're going to be scared. You're going to be alert. So in parasympathetic, you're resting. Think about it. When you are not doing schoolwork for nursing school, you're going to be at rest. You have time to go to the bathroom. You have time to drink things. You have time to digest things. Your pupils are going to be constricted because you're not having to worry about seeing anything. Everything's chill. So we have constricted pupils. We have digestion. We have peristalsis, which is moving food throughout our body. We have slow heart rate. We can go to the bathroom, which is peristalsis and digestion. All that's moving along. Okay, so when we're scared, our pupils are going to dilate. They're going to open up because we're like, holy crap, we want to see everything. Everything's going on. Our eyes are going to be wide. We're going to be ready to fight or flight. So sympathetic is fight or flight. And she will probably put that on the test. I feel like she's going to say that because there's been 1,000 questions about it. So we are not going to have digestion. We are not going to have peristalsis. And our heart rate is going to be fast. Because we're freaking out. So that's kind of jumping a little bit ahead. But so the autonomic nervous system is going to control the body's involuntary functions like heart rate, contractions of stomach and intestines, secretion of chemical compounds by glands. So we're going to bump over to 176, the cells of the nervous system. These are two major types of cells that are found in nervous tissue. We have neurons, which are the nerve cells, and we have glia, which are the supporting cells. So your big dude is going to be a neuron, and then the little dude that's going to help it is the glia. So if we come over to a neuron structure, each neuron consists of three parts. A main part called the cell body, one or more branching projections called the dendrites, and then one elongated projection known as the axon. So I'm going to try and draw this out to see if that'll help just a little bit. All right, so we have the cell body. I think the cell body is what they call it. Yes. All right, and then we have all these projections, and these are dendrites. All right, and then we have one long elongated piece, and this is the axon. Okay. And that is a better picture on 176. All right, so there's different types of neurons. So different types of this right here. And we have, go ahead over here. I'm going to erase this stuff. We can go back and look at that. All right. So there's different types of neurons. We have sensory, which are afferent. We have motor, which are efferent. And then we have interneurons. All right, so the sensory neurons carry impulses to the spinal cord and brain from parts of the body. So we're going to say body to brain. Okay, motor neurons carry impulses in the opposite direction, so they're going to go from the brain to spinal cord. So we're going to say brain to body. I'm so sorry about hearing y'all. I promise I'll write better than that. Interneurons conduct impulses from sensory neurons to motor neurons. So these are going to conduct from sensory neurons to motor neurons. So this is just a connection between these. So we'll say inter in the middle. All right, so that's the types of neurons. Now we have functions of glia or neuroglia. They don't specialize in transmitting impulses, but they are special types of supporting cells. So glia are like the cheerleaders. Their name is appropriate. It's derived from Greek, meaning glue, so it holds the functions together and protecting them. So basically, glia are cheerleaders. So all right. So glia perform many functions, including regulation of neuron function. They also act as a glue, and then they bring various functions of nervous tissues together in a coordinating whole. And we need to know this because the most common type of brain tumor is called a glioma, and this is a tumor inside of your glia. So if they're not being able to help, your body's not going to be able to function the way that it should properly. So there's central glia. Sorry. All right, so glia vary in shape and size. Some look like stars, so these are going to be our astrocytes. All right, and astrocytes have thread-like branches that attach to neurons. There's small blood vessels holding these structures close to each other, so these are going to connect to those. Along the walls of the blood vessels, astrocyte branches form a two-layer structure called a blood-brain barrier. So these form the BBB, and this is meant to help us, but it can also harm as well when we need to get something past that and we can't, because it's supposed to be protecting our body from chemicals, but we might need to get something past it and we can't. So that's not going to be very helpful. So it separates blood tissue and nervous tissue to protect vital brain tissue. So we're going to say this separates blood tissue and nervous tissue. Okay, so then we have microglia, and micro are going to be smaller. So we have our astrocyte, and then we have microglia, which are smaller. All right. So these are smaller than astrocytes. They remain stationary, but if they get inflamed or they're in degenerating brain tissue, they enlarge and they move about and move as microbe-eating scavengers around. They surround the microbe, draw them into their cytoplasm, and digest them, and then they clean up cell damage resulting from injury or disease. So we're going to associate microglia with phagocytosis. All right. Then next we have oligodendrocytes. These hold nerve fibers together and serve another and probably more important function. They produce a fatty myelin sheath that envelops the nerve fibers connected or located in the brain and spinal cord. The myelin sheath affects nerve conduction speed. So this, when we talked about a sheath before, it just has to do with like lubricating, has to do with keeping the train on the tracks. So oligodendrocytes are going to hold nerve fibers together. So I'm going to go ahead and erase some of this. Okay, we have oligodendrocytes. They're going to hold nerve fibers together. And these are, excuse me, they produce the fatty myelin sheath that envelops nerve fibers. So we are looking back at page 177. We see the central neuroglia and peripheral neuroglia. So central is going to contain astrocytes, microglia, and oligodendrocytes. So there's 3. These are all in the central peripheral. All right, and then so the myelinated sheath, excuse me, it affects conduction speed. I'm so sorry. All right, so myelin sheath faster is how we're going to think of that. So that is, we'll think of it like a superhero. Superheroes probably run faster when they have their elastosuits on, when they're all tucked together. So I know I'm going to move faster if I have my Spanx on. So that's the way I'm going to think about it, is the myelin sheath is like a Spanx on somebody. So then we're going to move down to central glia. Sorry. And then we have peripheral glia. These are going to be your Schwann cells. We had a lot of questions about those. Schwann cells. They're glia that also form myelin sheaths, but they only do it in the PNS. So they do myelin. I'm so sorry, y'all. Myelin sheaths, but in the PNS only. All right, so we have PNS, CNS, the different glias in each. Schwann cells are only going to do what oligodendrocytes do, but in the peripheral nervous system. Okay, so each oligodendrocyte can form part of the myelin sheath around several axons, but Schwann cells wrap entirely around only one axon. So neurons with myelin-wrapped axons are called myelinated fibers. So neurons that are myelin-wrapped with these Schwann cells are called myelinated fibers. And if you guys have any questions, please text me, please send something to the group, or please leave a comment and I'll stop. I'll try to go back and explain it the best way that I can. All right, so now we're going to go to nodes of Ranvier. They're gaps between adjacent Schwann cells. The outer wrap layer of the Schwann cell is called a neurolemma. So we have the Schwann cell. The outer wrapped layer is neurolemma. All right, and then nodes of Ranvier is the gap between adjacent Schwann cells. So these Schwann cells that are covered with neurolemma, this gap is called the node of Ranvier. And this is on page 178 at the top on the left-hand side. All right, so because there are no Schwann cells in the CNS, axons in the brain's and spinal cord have no neurolemma. So this area right here does not have neurolemma. The fact is significantly or clinically significant because neurolemma plays an essential role in regeneration of cut and injured axons. So you would say that this is kind of the skin over these Schwann cells, this neurolemma is. So if something happens to it, it's kind of like a little Band-Aid. So these are going to help with injured axons. So therefore, the potential for regeneration of damaged axons in the brain and spinal cord is far less when it's in this central glia area in the central nervous system. All right, so now we're going to bump over to nerves and tracts. So a nerve is a group of peripheral nerve fibers or axons that are bundled together. They act like strands of a cable. Peripheral nerve fibers usually have a myelin sheath because myelin is white. Peripheral nerves often look white. So the nerve is a group of peripheral nerve fibers or axons bundled. So when we bump back to page 176, we see that these axons are covered in Schwann cells, and a bunch of peripheral nerve fibers, a bunch of axons is a nerve. So this, we have it drawn out. All right, so 176, look at figure 9-2A, 9-2A, and that's going to show you what a nerve fiber and a nerve looks like. So peripheral nerve fibers usually have a myelin sheath because myelin is white. Peripheral nerves look white. Actually, no, a white guy, and his name is Myelin. Crazy. Okay. So anyways, that's a good way to remember that. Each axon in a nerve is surrounded by a thin wrapping of fibrous connective tissue called an endoneurium. So each axon in a nerve, let's see if we can go back and look at this, on page 176. Let's see if I can hold this up. This figure right here, this is one we're talking about where my thumb is. Okay, so let's see if we can do it this way. I'm really trying for y'all, I promise. Okay, so we see where the axon is and we see where the Schwann cells are. We see that a bunch of axons are neurons. All right, so now we're on the next page. There's a fibrous connective tissue called endoneurium, and this is surrounding the nerve. So groups of wrapped axons are called fascicles. Ooh-wee. All right, let me try and draw this out. I'm trying to explain it the best way that I can. I'm going to go ahead and erase this. I don't know about y'all, but I'm getting hot up here. Okay, so let's go ahead and draw this. We had our cell body. We had our dendrites. We had our axon. And then we have our Schwann cells in this particular picture. So Schwann cells. So, all right. A nerve is a group of peripheral nerve fibers bundled together like strands of a cable. So a nerve is more than one axon. All right. Each axon in a nerve is surrounded by a thin wrapping of connective tissue called endoneurium. So each of these little axons has, it's like a jacket around it. And these are endoneurium. Okay. And then groups of these wrapped axons are called fascicles. So more of these, the ones that are wrapped are called fascicles. All right, each fascicle is surrounded by a thin fibrous perineum. So all of these that are covered is covered again by perineum. I really hope that that's not hard to keep up with. I promise I'm trying. Okay, a tough fibrous sheath called the epineurium covers the whole nerve. So the epineurium is what's going to cover this entire area of bundled axons, which is the nerve. Does that make sense? Is that good for everybody? Please let me know before I keep going because I don't want to confuse anybody. And we probably won't have to go that deep into it. We're probably just going to have to know the terminology. But if I see that, it helps me understand it. Everybody good? All right, I'm going to go ahead and erase this. All right. So we're going to get into a different, kind of change gears a little bit. And actually, change my marker color. All right, so nerve signals, reflex arcs, and neuron pathways. All right, so hormones are the only other kind of signal that the body can send. Oh, I'm sorry, I bumped down just a little bit. Bundles of axon in the CNS are called tracts. So bundles of axon in the PNS. CNS are neurons. Nope, nerve, sorry. And then in the CNS, I'm trying. Bundled axons in the CNS are called tracts. How can they just all have the same name? All right. Okay, so these are also myelinated, and they form white matter of the brain and spinal cord. Brain and spinal cord tissue composed of cell bodies and unmyelinated axons and dendrites are called gray matter. So the ones that are myelinated are white. And then non-myelinated are gray. And this isn't super significant, but plays a role later. All right, so now we're going to move down to nerve cells, reflex arcs, and neuron pathways. So hormones are the only other kind of signal that can send, and they travel much slower than nerve signals. Hormones can move from one part of the body to another through circulating blood. So nerve impulses are a lot faster. So we are going to say, all right, so nerve impulses are like Amazon overnighting. And then hormones are snail mail. It's going to take a while for a hormone to set in, just like snail mail would take. If you put a post stamp on something and send it in the mail, it's going to take a while. These nerve impulses are going to be like Amazon overnight, and those are pretty quickly. And you want it to be that way because if you put your finger on something that's hot, you don't want to take it 5 minutes for you to pull your hand away and then you burn yourself. You want it to be instant. You want these nerve impulses so that it's instant that you pull your hand away and you're less likely to get harmed. This is the whole point of having senses, so that your body can be protected, so that you can maintain homeostasis. So nerve impulses are also called action potentials, and they travel over trillions of routes made up of neurons because they are cells that conduct impulses. So the routes traveled are sometimes called neuron pathways. So the picture that we had with the cell body and the dendrites and the axon that came off, these axons are going to go everywhere. So the basic type of neuron pathway is called a reflex arc, and it's important to the nervous system functioning. The simplest kind of reflex arc is a 2-neuron arc. So 2-neuron arc is the simplest, and this is a okay, yep, so now we're talking about reflex arcs. The simplest is a 2-neuron arc because it consists of only 2 types of neurons, sensory and motor. All right. And then a 3-neuron arc is the next simplest kind. It consists of 3. We have a sensory and motor. All right, so then we have 3 NA. And this is sensory and motor plus interneurons. Okay, so that's the only difference between those two. So structure of reflex arcs, they're like a one-way street. They allow impulse conduction, but only in one direction. So it's just sending things out. It's not receiving anything. Impulse conduction normally starts in receptors. The receptors are the beginning of dendrites of sensory neurons. So I believe we just looked at this in the other chapter. I think I'll get back to it. Yes, so they're kind of like the neuromuscular junction in the muscles. Receptors are going to be the beginnings of dendrites of sensory neurons. So they're often located far from the spinal cord, and examples, tendon, skin, or mucous membranes. So let's see, down here in figure 9-5, the stretch receptors are stimulated when muscles are stretched as a result of a tap on the patellar ligament from a rubber hammer that's used by a physician to elicit a reflex during a physical examination. So the nerve impulse that's generated is a neurological pathway, and its ultimate knee-jerk effects are an example of the simplest form of 2-neuron arc. So in a knee reflex jerk, only sensory and motor neurons are involved. So it's going to pick up something and it's going to give back. That's just 2. Okay, so nerve impulses that is generated by stimulation of the stretch receptors travels along the length of a sensory neuron's dendrite to its cell body called the dorsal root ganglion or posterior root ganglion. Ganglion is a group of nerve cell bodies located in the PNS, and this ganglion is located near the spinal cord. Each dorsal root ganglion contains not one cell body, but hundreds of them. The axon of a sensory neuron travels from the cell body to the dorsal root ganglion and ends near the dendrites of another neuron located in the gray matter of the spinal cord. So microscopic space that separates the axon ending of one neuron from dendrites in another neuron. This gap serves as a junction between nerve cells called a synapse. The nerve impulse stops at a synapse. Chemical signals are sent across the gap, and a new impulse continues along the dendrite, cell body, and axon of the neuron motor. The motor neuron axon forms a synapse with a structure called an effector, an organ that puts nerve signals into effect. Effectors usually are muscle glands, muscle contraction, muscle secretion, and kinds of reflexes operated by these effectors. So I'm going to kind of explain that in just a minute. I'm going to be able to draw that for you. So basically, the end of a dendrite is going to be, or the beginning of a dendrite is going to be your receptor, and these are going to send out an impulse. So example of two neuron arc is the knee jerk. So if you're getting hit on the knee with a hammer, you're getting hit, you're giving off a reflex. This is 2. So then it tells us that, let's see, okay, nerve impulse is generated by stimulation of stretch receptors, travels along the length of a sensory neuron's dendrite, which are the long figures coming off of the cell body. Let's see, I'm losing my place. To its cell body located in the dorsal root ganglion or posterior root ganglion. A ganglion is a group of nerve cell bodies located in the PNS. So the circle that we drew, the cell body, a group of these are ganglion, and this is only in the PNS. So the ganglion is located near the spinal cord. Each dorsal root ganglion, dorsal root ganglion consists not one sensory neuron cell body, but hundreds of them. All right, so a synapse. The best way that I saw to view that was on the Evolve Learning. It had a very, very short video, and it was really good. I mean, it was literally just seconds long, but basically, if you hold your fists together, you can use this as an example. So let's see if I can do this. All right, so this is an axon. And this is an axon. All right, there's a space. All right, I'm going to do the best I can here. This is the synapse right here. This is what we're talking about on 179. The space between 2 axons. All right, and it's a microscopic space. It's super tiny. It's like something millionth of a space. It's super, super small. I'm sure we're about to run into that in just a second. Okay, the nerve impulse stops at the synapse. So if there's a nerve impulse coming down, it's going to stop. And chemical signals are sent across the gap, and then a new impulse continues along the dendrite cell body and the axon of a motor neuron. So motor neuron axon forms a synapse with a structure called an effector, an organ that puts a nerve signal into effect. So these are, once there's a signal sent across, it's going to trigger an effector, which is like a hormone that needs to be secreted, or a muscle that needs to be contracted, or a gland that needs to be secreted. I've already said that. Okay, so that's all this is just like a highway for these to be moving. So I'm going to go over to reflex responses. An involuntary response to impulse conduction of a reflex arc is called a reflex. So when you get hit on the knee with that hammer, you don't control whether or not your knee goes up. That's just something that happens. That's a reflex. So it's an impulse conduction by a reflex arc, and that's how it causes it to happen. So there's an interneuron shown in figure 9-5. Some reflexes involve 3 rather than 2 neurons, and these more complex types of responses are interneuron, in addition to sensory and motor neuron, is involved. So there's 3. There's an interneuron, sensory, and a motor neuron. In 3 neuron reflexes, the end of a sensory neuron's axon synapses first with an interneuron before the chemical signals are sent across to the second synapse. So in this case, this is a 2-way reflex arc. And then for a 3-way, we're going to do 3-way over here. 2 is going to have nothing in this synapse, nothing in this gap. 3 is going to have interneuron. Okay. All right, so in a 3-neuron reflex, the end of a sensory neuron's axon synapses with the first interneuron before chemical signals are sent across the second synapse, resulting in a conduction through the motor neuron. Excuse me. For example, application of irritating stimulus to the skin of the thigh initiates a 3-neuron reflex response that causes contraction of muscles to pull the leg away from the irritant. A 3-neuron arc reaction called a withdrawal reflex. So I think we probably just need to know the bolded term for that. All interneurons lie entirely within the gray matter of the brain or spinal cord. So all of these are going to be in the brain or spinal cord. Gray matter forms the H-shaped inner core of the spinal cord because there's a presence of interneuron. 3-neuron reflex arcs have 2 synapses. However, a 2-neuron reflex arc only has a sensory neuron and a motor neuron with 1 synapse between them. So I would also go back, please go back and look at your Evolve Learning because it's free with your book and it's very helpful, and it shows us that if you're looking down, if somebody was looking down my spine into it, that's what they're talking about when they say that there's like an H shape. It kind of looks literally like an H, and this is how that is controlled. So this is a gray matter that forms that H shape. Because of the, let's see. Oh, so in figure 9-5, it says observe that its dendrites and cell body, like those of an interneuron, are located in the spinal cord as gray matter. So all of those are found in that H. However, the axon of this motor neuron runs through the ventral root or anterior root of the spinal nerve and terminates in the muscle. I can't draw that. I can't explain it, but it's in your Evolve Learning. Please watch those. So nerve impulses, definition of a nerve impulse. So let's see. Okay. So there's okay, so it's saying you can visualize a tiny spark sizzling its way down a fuse. Nerve impulses do not continually race along every cell surface. They first have to be initiated by stimulants or a change in the neuron's environment. Pressure, temperature, and chemical changes are usual stimuli. So we'll keep this here. We'll keep that there because it's going to be helpful to explain it. So mechanism of a nerve impulse. The membrane of each resting neuron has a slight positive charge on the outside and a negative charge on the inside in the state called polarization. So we have a slight positive charge on the outside and a negative charge on the inside. And this is called polarization. This occurs when there's normally an excess of sodium ions on the outside of the membrane. So we have Na plus. All right, so this state right here is polarization. All right, so just keep in mind as we're doing this, it kind of comes to mush. It kind of gets a little mumbo jumbo, but just think of it as we're trying to reach a goal, and our goal is to get a nerve impulse. So the goal is we're trying to pull our hand away from something really hot. So this is what has to happen for us to get to that point. So when a section of a membrane is stimulated, its Na channel suddenly opens up and the Na ion is rushed inward. So when this is stimulated, there's some kind of impulse coming down, this is going to move into the membrane. The inside of the membrane temporarily becomes positive and the outside becomes negative. And this is depolarization. Okay. All right, so depolarized section of the membrane immediately recovers in a process called repolarization. All right, so then okay. All right, so basically this is going to go back to its normal self, and then that would be called repolarization. But this Na is already in this next synapse, or not next synapse, in the next axon. So this is going to continue on. So the conduction of a nerve impulse, the impulse or action potential cannot go backwards during the brief moment of repolarization. So when that swaps back to where it was to begin with, this Na can't be like, oh, never mind, and pull back. It's got to keep going. That's the nerve impulse. It's just one swift motion. So the self-propagating wave of electrical disturbance and nerve impulse continues into one direction across the neuron surface. So nerve impulses are called action potentials, and you know that. Nerve impulse, action potential. Okay. Because each one is different in charge, it's called electrical potential. It usually triggers an action by the cell, but in this case, a transmission of the impulse itself. So if a traveling impulse encounters a section of a membrane covered with an insulated myelin, it simply jumps around it into the next gap in the myelin sheath, and this is called saltatory conduction. So when there is see how best I can do this for you. All right, so if the traveling impulse encounters a section where there is a membrane covered in insulated myelin, this is insulated myelin, it's going to jump around. And this is called saltatory conduction. Okay. And this type of impulse travel is much faster than it is in a non-myelinated section, because when we think of myelin, we think of our Spanx. We're going to move faster in our Spanx because our fat's not jiggling around. So the purpose of saltatory conduction is to just make this faster. All right, so now we're going to talk about synapses. I'm going to kind of start over with this diagram because it's pretty much ruined at this point. Y'all leave me some comments. Tell me what you're wanting to hear, and if I'm hitting stuff well enough, or if I need to go back and explain something else, I don't mind. It's only 5:30, y'all. All right. So the structure and function of a synapse, transmissions of signals from one neuron to the next cross the synapse. So this is our neuron, which is a bunch of axons. Let us not forget that. So neuron. This is another neuron, and we're trying to have an impulse. We want something to move down. We want something to happen. So this is our synapse, which is our space. Think synapse space. By definition, it's a place where impulses are transmitted from one neuron called the presynaptic neuron to another neuron called the postsynaptic neuron. So this is presynaptic. And this is postsynaptic, first and second. All right, so 3 structures make up a typical synapse: a synaptic knob, a synaptic cleft, and a plasma membrane of the postsynaptic neuron. So the synaptic knob is going to be this area right here. And then a synaptic cleft is going to be right here, which is synapse. All right. And then, all right, so I'm trying not to lose my place. And then we have the plasma membrane of the postsynaptic neuron. So this is your plasma membrane right here, what it's trying to get through, which is, we remembered from the cell chapter. Okay. So synaptic knob is a tiny bulge at the end of the terminal branch of a presynaptic neuron's axon. So that's what this is right here. Each synaptic knob contains many small sacs or vesicles, and each vesicle contains, so basically this is shaped a little bit, I'll shape it a little bit better. This looks like a knob. This is what it looks like better in the picture. I'm just getting a little lazy drawing it. So this knob is this bottom part. This is going to contain a bunch of little vesicles, and inside of the vesicles is a very small quantity of chemical compounds called neurotransmitters. So these are the next paragraph or so. But the neurotransmitters are what needs to be released. So when a nerve impulse arrives at the synaptic knob, neurotransmitter molecules are released from the vesicles into the synaptic cleft. So these are going to come out. And now these are in here floating around. All right. So the synaptic cleft is the space between the synaptic knob and the plasma membrane of the postsynaptic neuron. and it's an incredibly narrow space. It's only 2/millionths of a centimeter in width, and it's the synaptic cleft is filled with extracellular matrix that holds the synaptic structure in place. So this is filled with extracellular fluid. All right, so the plasma membrane of the postsynaptic neuron has protein molecules embedded in it in opposite each synaptic knob. So whatever is here is going to be opposite of here. These serve as receptors to which neurotransmitter molecules bind. These bindings can initiate an impulse in the postsynaptic neuron by opening ion channels in the postsynaptic membrane. Each type of neurotransmitter can only bind to a receptor that chemically fits with it, much as the enzyme fits with the substrate molecule. The characteristics of neurotransmitters and receptors allow for very precise body function. So after an impulse conduction by a postsynaptic neuron is initiated, neurotransmitter activity is rapidly terminated. Either one or both of two mechanisms causes this. Some neurotransmitter molecules are transported out of the synaptic cleft back or into synaptic knobs. Other neurotransmitter molecules are broken apart into inactive compounds by specific enzymes in the extracellular matrix of synaptic cleft. I'll try to back that up just a little bit. These neurotransmitters stay in the synaptic knob, and then they move out into the synaptic cleft, and then they're going to kind of, what was the word that they used? Break apart into inactive compounds. So now we're going to talk about neurotransmitters, and these are the chemicals in which the neurons communicate. So there's a lot going on in these things. As previously noted, trillions of synapses in the CNS, presynaptic neurons release neurotransmitters that assist, stimulate, or inhibit postsynaptic neurons. At least 30 different compounds have been identified as neurotransmitters, and they are not distributed randomly through the spinal cord and brain. Instead, specific neurotransmitters are localized in discrete groups of neurons and released in specific pathways. So, for example, a substance called acetylcholine is released at some of the synapses to the spinal cord and neuromuscular nerve muscle junctions, which we talked about earlier. And then we have norepinephrine, dopamine, and serotonin. So I'm going to go ahead and erase this. So those little vesicles filled with the neurotransmitters is what we're talking about now. We're talking about neurotransmitters and their purpose. So all right, so the different kinds of neurotransmitters, we have acetylcholine, which we talked about earlier, and then we have norepinephrine, we have dopamine, and then we have serotonin. All right, they belong to a group of compounds called amines. All right, which play a role in sleep, motor function, mood, and pleasure recognition. So sleep, motor function, mood, and pleasure recognition. So you can think of me when you take the test tomorrow. I take Zoloft, and this is a serotonin uptake inhibitor. So if I'm struggling with depression and other things going on, serotonin is going to it's more neurotransmitters in my body triggering serotonin to allow me to be happy. So that's what I think of when I think of those. Dopamine is kind of like that. It's like a high feeling, if that makes any sense. It's just a relaxed state. There's a lot of good going on in your body. This is also like when you relax and you're sleeping. This has to do with your mood. And then there's two morphine-like neurotransmitters called endorphins and enkephalins, and these are released at various spinal cord and brain synapses in the pain conduction pathway. These neurotransmitters inhibit conduction of pain impulses, and they are natural painkillers. So when we talk about endorphins and enkephalins, I think of ears. If you go to get your ears pierced, you get a piercing on this side and a piercing on this side, kind of freaking out. First time you got your ears pierced, you're like 5 years old. You don't know how that's going to feel. So they come in with the thing that declares, which was not a good idea. They're going to pierce this ear after you're panicking, and you're going to say, oh, it wasn't that bad. That didn't feel that bad. You kind of get this hot flush feeling over you. That's because all your endorphins are running to this area where you get it pierced. The next part is not going to be as fun when you get the next ear pierced. That's going to be a lot more painful because all of your endorphins and enkephalins are already on this side, rushing to this for your pain receptors or natural painkillers. So the second one's going to be a lot worse than the first because that's where all of these are still concentrated. So that's what I think of as enkephalins and endorphins, ears. It's a natural painkiller. Very small molecules such as nitric oxide or NO have an important role in neurotransmitters. Unlike most other neurotransmitters, this diffuses directly across the plasma membrane of neurons rather than being released from vesicles. So, like we had it in our axons that we showed, and then the little vesicles filled with these neurotransmitters. Small molecules like nitric oxide do not diffuse across plasma. They just are. They release directly across the plasma instead of being diffused first. So they just come straight out. Okay, so now we're going to go over to central nervous system, but I'm going to say that we go ahead and take like a 5-10 minute break. And I know some of us are getting ready to go to church. That's totally fine. This should be uploaded as soon as it's finished. So if you're missing something, you should be able to come back and see it and look at it when you're back. So let's go ahead and take a 5-minute break. Let's come back at, let's come back at 5:45. Let's do that. And then we will get started on the central nervous system. All right, so if you're still logged in, will you go ahead and comment so that I know you're here and ready to start? I had to go get me a cookie, y'all. All right, so now we've kind of gotten past the hard part, which is all the little pieces that make up the big puzzle. Hey, Valerie. Now we're going to move on to page 184, and this is where we get into the central nervous system, and then it kind of gets a little bit easier here. This is more of the memorization part rather than having to try and tie things together. So the CNS, it's two major structures, the brain and spinal cord, like we said. So we have brain and spinal cord. The brain's protected by in the cranial cavity of the skull, and the spinal cord is surrounded by the spinal cavity in the vertebral column. So you could say that the PNS takes care of the CNS. So it's protected by three membranes called meninges, and that's going to be discussed a little bit later. So we have divisions of the brain. We have four main divisions. So we have brainstem. And I don't know about y'all, but I like to see pictures of it. So I'll put those pictures on the website. And that helps me to be able to understand what parts of the brain do what. So if you're just talking to me about it, I'm not going to understand it. And then we have cerebellum. And then we have diencephalon. And then we have cerebrum, which is the biggest part. All right. So the brainstem contains three things, and the brainstem literally looks like a stalk of broccoli without the top part, and it's in three different parts. And we have the midbrain, which is going to be towards the middle of the brain, and then we have the pons in the middle, and then we have medulla oblongata at the bottom, and apparently that is Miss Mickey's favorite word, so that's definitely going to be on the test. I would definitely know that. All right, diencephalon has the hypothalamus, the thalamus, and pineal gland, which is my favorite. All right, and then we said the cerebrum is the biggest part, and this has the two halves of the brain, the two halves of the top, so it's the biggest part. All right, so we're going to bump over to 185 under brainstem. So the lowest part of the brainstem is the medulla oblongata. Immediately above that is the pons, and above that is the midbrain. The medulla oblongata is an enlarged upward extension of the spinal cord. So below this is our spinal cord. And it lies just inside the cranial cavity above the large hole in the occipital bone called the foramen magnum. So back here, this is the back of our brain, which is the occipital bone. The pons bulges out a little bit more than the medulla, forming the bridge to a narrower medulla oblongata. So this is just a little bit bigger, is all that it's telling you. All these parts of the brainstem function in two-way conduction paths. So sensory fibers conduct impulses from the spinal cord to other parts of the brain. And then motor fibers conduct impulses from the brain down to the spinal cord. So we have motor here, and then we have sensory here. Okay. All right, so in the brainstem, small bits of gray matter mix closely with intricately white matter to form reticular formation, which means net-like. So in the spinal cord, gray and white matter do not intermingle. Gray matter forms the interior core of the spinal cord, white matter surrounds it. So in the spinal cord, we have gray on the outside and then white on the inside. And then here, it kind of mingles a little bit, but they don't mix in the spinal cord. So many important reflex centers lie in the brainstem, such as cardiac, respiratory, vasomotor centers, collectively called vital centers. For example, they're located in the medulla, and impulses from these centers control heartbeat, respiration, blood vessel diameter, which is important in regulating blood pressure. So this is the bulk of it. It's a teeny tiny little area, but it has a lot to do with it. All right, so now we're going to bump over to cerebellum. So it's the second largest part of the human brain and lies under the occipital bone of the cerebrum. So if you're looking at a picture of the brain, and try to do my best to draw this. All right. We have, that is, oh, that's terrible. I'm so sorry. This little area back here is cerebellum. And this is the brainstem. Okay, so the cerebellum is the second largest part of the human brain, and it lies under the occipital bone of the cerebrum. In the cerebellum, folded gray matter composes a thin outer layer and forms a large surface area of neuron connections that allow for huge amounts of information processed. So white matter tracts form most of the interior. Notice that these tracts branches have a tree-like pattern called an arbor vitae, or living tree. So the little projections that are off of there is that's what this is talking about. So most of our previous knowledge about cerebellar functions has come to observing patients who have some sort of disease of the cerebellum, and from animals that have had a cerebellum removed. From these observations, we know that this plays an essential part of production of normal movement. So a few examples: a patient who has a tumor of the cerebellum frequently loses balance and topples over, might feel drunken when he walks. He cannot coordinate this muscle normally, and he might complain, for an instance, that he's clumsy, that everything he can't drive a nail or draw a straight line. So with the loss of normal cerebellar functioning, he's lost the ability to make precise movements. So this has to do with our walking, grabbing things, fine motor skills, things like that. So let's see, most obvious functions of the cerebellum are to produce smooth, coordinated movements, maintain equilibrium, and sustain normal posture. So recent studies using new brain imaging methods show that they have far more functions than earlier observed, perhaps overall over-coordinating function for the whole brain, so that we kind of think that this has to do with all of this. The diencephalon is small, but it's important, and it has three major functions. We have the hypothalamus, thalamus, and pineal gland. So the hypothalamus is located below the thalamus, and the posterior pituitary gland, the stalk that attaches to the undersurface of the brain. Areas of gray matter located on the side walls of fluid-filled space called third ventricles are extensions of the hypothalamus, and identified to okay, it's telling us to look at figure 9-10 for these glands. So hypothalamus is small, but it's one of the most significant parts of the brain, and it's a contribution to healthy survival. Impulses from neurons, whose dendrites and cell bodies lie in the hypothalamus, are conducted by their axons and neurons located in the spinal cord, and these impulses are then relayed to muscles and glands over the body. So the hypothalamus exerts major control over virtually all internal organs. So among vital functions, it helps to control heartbeat, constriction and dilation of blood vessels, and contractions of stomach and intestine. So this part is going to largely be affected and parasympathetic and sympathetic, because it controls constriction and dilation of your blood vessels. It has to do with your stomach, your intestines, has to do with your heartbeat. All those things are going to be affected in sympathetic and parasympathetic systems. Sorry. All right. So neurons in the hypothalamus function in a surprising way. They make hormones that the posterior pituitary secretes into the blood. So one of the hormones is an antidiuretic hormone, or ADH, and affects the volume of urine excreted. The hypothalamus also plays a role in maintaining the body's water balance. So if you look over at, well, there is a chart somewhere. Maybe we jumped past it. I know we didn't. Sorry. Oh, okay. When it comes to, this has to do with it, the endocrine chapter, sorry. All right, so the antidiuretic hormone has to do with urine excretion, and then it also functions in, I'm sorry, I'm losing my place. All right, so a function in endocrine or ductless glands. So endocrines go straight into your bloodstream, and exocrine go through ducts. So when we're dealing with the hypothalamus, we're dealing with ductless glands. So their axons secrete chemicals called relaxing hormones into the blood, which carries them to the anterior pituitary gland. So releasing hormones, as their name suggests, control the release of certain anterior pituitary hormones. These in turn influence the hormone secretion of other endocrine glands. So the hypothalamus indirectly helps to control the functioning of every cell in the body. So the hypothalamus is going to create these, but then it's going to kind of send them off for packaging. So that would be, I have some mail that I want to send out. I'm going to send it to the post office, is the hypothalamus, and then the hypothalamus is going to send it out to where it needs to go, to somebody's mailbox. So the hypothalamus is a crucial part of mechanism for maintaining body temperature. Therefore, marked elevation in body temperature in the absence of disease frequently characterizes injuries or other abnormalities of the hypothalamus. In addition, it's involved in regulation of water balance, sleep cycles, and control of appetite, and many emotions involved in pleasure, fear, anger, sexual arousal, and pain. So also in the diencephalon, we have the thalamus. In this, each enlarged end of the dumbbell lies at the lateral wall of fluid-filled chamber called a third ventricle. The thin center section of the thalamus passes from left to right through this ventricle, which is discussed more in detail later. It's composed of chiefly of dendrites and cell bodies and neurons that have axons extended up towards the sensory area of the cerebrum. So it performs the following functions. There's quite a bit. It relays sensory information, associates sensation with emotion, regulates a level of consciousness, and participates in motor reflexes. Its neurons relay impulses from the cerebral cortex to the sense organs of the body, and almost all sensations are accompanied by a feeling of some degree of pleasantness or unpleasantness. So the way that these pleasant and unpleasant feelings are produced is unknown, except for they seem to be associated with the arrival of sensory impulses to the thalamus. So we don't know why we feel a certain way. We just know that it has to do with the thalamus. Regulates level of consciousness that plays a part in so-called arousal or alerting mechanism that keeps us awake. So when you go in to check and see if your patient is AAO, you're going to know that that's associated with the thalamus. Also participates in motor reflexes that plays a role in mechanisms to produce complex reflex movements, which we talked about before, like a reflex arc. So we're going to go to pineal gland. This is the last one in the diencephalon. So posterior to the thalamus is a tiny mass protruding. It kind of just looks like a little chestnut. It's the pineal gland or pineal body. And it receives sensory information about the strength of light seen by the eyes and adjusts its output of the hormone melatonin. So melatonin is known as a timekeeping hormone because it helps to keep the body's clock on time with daily, monthly, and seasonal cycles of sunlight and moonlight. So you can buy this. I do. When I get really tired and I know I get a chance to sleep and I don't have a test coming up, I will take melatonin and I will sleep for like 13 hours straight. So it's already produced in your body and it's non-habit forming. But this is just to enhance what's already being produced in the pineal gland. The pineal gland is melatonin. It's the timekeeper, and we will have to know that. So the cerebrum is the biggest part. It makes up the left and right hemispheres. So you might notice that there's ridges and grooves. The ridges called convolutions are gyri, and the grooves are the sulci. So the gyri would be the bigger part. This is the convulsion. It convulsions out. And then the grooves are the sulci, so the parts in the middle are the sulci. So the deepest sulci are called fissures. The longitudinal fissure divides the cerebrum into left and right halves or hemispheres. So what this is talking about is these little areas. Okay, so if this is a bump in the brain, this like it's kind of bumped up a little bit, these are going to be the gyri, kind of gyrating out. All right, and then the sulci is going to be between them, kind of sulk down into it. And then the deepest one, the deepest sulci is the middle part right here that separates the brain into left and right halves. And they're almost completely separated except for an inferior central band called the corpus callosum, which is made up of white matter tracts. So two deep sulci subdivide the cerebral hemisphere into four major lobes. It's going to separate into four major lobes. Into numerous convolutions. The names are lobes are named for the bones that lie over them. So you have a frontal, you have a parietal, you have your temporal, and then you have your occipital in the back. The thin layer of gray matter called the cerebral cortex, made up of neuron dendrites and cell bodies, forms the surface of the cerebrum. White matter, made up of a bundle of nerve fibers or tracts, composes most of the interior of the cerebrum. However, within this white matter is a few islets of gray matter known as basal nuclei or basal ganglia, whose functioning is essential for producing autonomic movements and postures. So the function of the cerebrum is neurons of these various structures continually bring impulses to cerebral neurons and also continuously carry impulses away from them. So if all of the neurons were functioning normally and the cerebral neurons were not functioning, there were some things you couldn't think, you couldn't use your free will, you couldn't remember anything that has ever happened to you, you would not be able to decide to make the smallest movement, nor could you make it. You would neither see nor hear, you wouldn't experience any of the sensations that make life the way that it is, and nothing would anger or frighten you, and nothing would bring you joy or sorrow. You would, in short, be unconscious. So this has to do with, I'm sorry. Y'all, I'm getting so tired of the cerebrum. Okay. So if this is not functioning correctly, you're not functioning correctly. You're probably going to be in a coma. So these terms sum up the major cerebral functions: consciousness, thinking, memory, sensations, emotions, and willed movements. And then I would look at page 188. This has a better breakdown than I could draw for you. So injury or disease can destroy the neurons, and a common example is a CVA or cerebrovascular accident. So this has to do with like a cutoff of blood flow in the brain or some kind of hemorrhage, some kind of damage to it. And so if there's damage in the right side of your brain, it's going to affect the left side of your body. If there's a damage to the left side of your body, it's going to affect the right side, or the left side of your brain is going to affect the right side of your body. So if I've had a stroke on the right side of my brain, then I'm not going to be able to move the left side of my body. It controls the opposite half. And there's some pretty cool studies that they have where they have given someone a test on one half of their body, and it's really confusing but super cool because everything is controlled by the opposite side. So that's something pretty cool to look into if you're interested in it. But a CVA is just a cutoff of blood supply in the brain, and it's just not going to be able to function properly. Let's see. The temporal lobe's auditory areas interpret incoming nervous signals from the ear as specific sounds, and the visual area of the cortex and the occipital lobe help you to identify specific images. The localization of function explains the specific symptoms associated with an injury to localized areas of the cerebral cortex after a stroke or traumatic injury to the head. So now we're going to go down to spinal cord. And let me know if there's any questions. If you guys have any questions or anything that you want me to specifically go over, you can comment, and I'll go back if you need me to. Definitely, I would know the chart 9-2. That's one I know she said we needed to go over. Whether you have to do it in a picture or whether you have to remember that chart, that's going to be important. She wants to know that you know what each part of the brain does. All right, so we're going to go down to spinal cord structure. It's about 17 to 18 inches long, and it lies inside the spinal column in the spinal cavity and extends from the occipital bone down to the bottom of the first lumbar vertebrae. So I don't think we're going to need to know that. It consists of gray matter and is composed mainly of dendrites and cell bodies of neurons. Columns of white matter form the outer portion of the spinal cord, and bundles of myelinated nerve fibers or spinal tracts make up the white column. So we talked about that a little bit before. Spinal tracts is the white matter. Spinal cord tracts provide a two-way conduction path to and from the brain. So ascending tracts conduct impulses up the spinal cord into the brain, and descending tracts conduct impulses down the spinal cord. So descending down, ascending is going to be up. Tracts are functional organizations in that all axons composing of one tract serve one general function. So, for instance, fibers of the spinothalamic tract serves a sensory function. They carry impulses that produce sensations of crude touch, pain, and temperature. And so this is going to be discussed a little bit later. Let's see. Other ascending tracts are shown in Figure 913. They include gracilis and cuneatus tracts, which transmit sensations of touch and pressure up in the brain. And the anterior and posterior spinocerebellar tracts, which transmit information about muscle length to the cerebellum. All right, let's see. So the functions of the spinal cord. This says to think of a hotel telephone switching system. So there's a guest in one room that's trying to reach another. So they're going to be sent through a switching system. A message is traveled to the switching system. The system is routed through a proper path, and then the message is traveled out of the switching system towards the next room. So basically, the spinal cord is just a highway for messages. Reflexes that result from conduction over arcs whose centers lie in the spinal cord are called spinal cord reflexes. And two common kinds of spinal cord reflexes are withdrawal and jerk reflexes. So an example of withdrawal reflex is pulling one's hand away from a hot surface, and a familiar knee jerk is an example of a jerk reflex. Let's see. The spinal cord tracts carry impulses to and from the brain. Sensory impulses travel up to the brain in ascending tracts, and motor impulses travel down from the brain in descending tracts. So we'll go ahead and put that up here. All right, so we have our, I need to erase this. All right, so you have your spinal cord. All right. All right, so sensory impulses travel up to the brain in ascending tracts. And then I'm descending. All right. Sensory travels up and motor travels down. All right. So the next thing we're going to talk about is meninges. These are pretty much memorization of the bolded term. So nervous tissue is not sturdy. Even moderate pressure can kill nerve cells. So nature safeguards the chief organs made up of this tissue, the spinal cord and brain, by surrounding them with tough, fluid, cushioned sets of membranes called meninges. And these are in figure 9-14. So they're kind of tube-like coverings around the spinal cord and line the bony vertebral forum of the vertebrae that surround the spinal cord. So there's dura mater, which is the tough outer layer that lines the vertebral canal. And then there's pia mater, which is the innermost membrane. And then there's arachnoid mater, which is the membrane between the dura and the pia. So arachnoid resembles a cobweb, and that word actually means cob-like. So let's see if I can draw a picture of this for you guys. It's really not something that we need to necessarily know like that, but sometimes it's good to see it. Okay, I got it. All right, so in this spinal cord, we have, this is just a closer look at the spinal cord. So there's 3 layers to this. If we're breaking it down, there's like an outer layer, there's a more inner layer, and then a more inner layer. All right, and these are just around, when somebody says meninges, I'm thinking of something completely different than what it is, but the outer is the Dura, the middle is the pia, and the inner is arachnoid. So we have dura, pia, and arachnoid. So these are basically just safeguards to keep this to keep your spinal cord safe, because if that is tampered with, broken, injured, you are going to be it's not going to be good. Okay, so arachnoid means a cobweb-like, and it comes from arachne, the Greek word for spider. Meninges that form the protective coverings around the spinal cord also extend up and around the brain to enclose it completely, which is going to be that look around a brain if you've ever watched a autopsy. All right, so the meninges are inner soft coverings of the CNS. They are surrounded by the hard bone of the skull and vertebrae, forming a highly protective shield from injury. All right, so basically these are just, we'll call them bulletproof vests. There's 3 of them, so they're going to be very, very safe. Sorry, my child's downstairs screaming. All right, so we have cerebrospinal fluid spaces. Fluid filled the subarachnoid spaces between the pia mater and... Yes, McKenna Ray? Got my paper. No, I didn't. I said Dad needed to get it, baby. Okay. Cerebrospinal fluid. What, McKenna? Excuse me. Sorry. Daddy, go downstairs on video. Lord jeez. All right. So the fluid that fills the subarachnoid spaces, which is the, I'm sorry, now I'm lost my train of thought. Between the pia mater and arachnoid and the brain and spinal cord is called cerebrospinal fluid. So you'll hear this a lot when you get to like the reproductive system, when you're getting ready to have a baby. That's where they're going to give you an epidural to numb your body, because this is what's running up and down your body. All right, so CSF also fills spaces in the brain called cerebral ventricles. And there's a figure 915, which is not there. Oh, okay. It's on page 192. So they lie deep inside the brain, and there are 2 lateral ventricles. So one lies on the right side, and the other is on the left side. Also, the left or right half of the cerebrum is the largest part of the human brain, so it's just a little bit bigger than the other. All right, so CSF forms continually from fluid filtering out of the blood into a network of brain capillaries known as choroid plexus and into the ventricles, which is the part that's inside of your brain that we talked about. So it's only one of the body's circulating fluids. It seeps from the lateral ventricles in the 3rd ventricle and flows through the cerebral aqueduct. Most of the CSF moves through tiny openings from the 4th ventricle into the subarachnoid space near the cerebellum, and some of it moves into a small tube-like central canal of the spinal cord and then into the subarachnoid spaces. Then CSF moves leisurely down and around the spinal cord and up around the brain in the subarachnoid spaces of their meninges and returns to the blood in the veins of the brain. So remembering that this fluid forms continually from blood, circulates, and is reabsorbed into the blood can be useful. It's also to help you understand abnormalities. So if a person has a brain tumor that presses on this aqueduct, this blocks the way for the return of CSF to the blood. And the fluid continues to form but can't drain away. It accumulates in the ventricles or the meninges, creating enough pressure to damage or deform the nearby soft nervous tissue. So it's very important that we don't have all that fluid on our brain. So if you do have all the fluid on your brain, this is called hydrocephalus, or water of the brain. A form of treatment involves surgical placement of a hollow tube or catheter through the blocked channel so that CSF can drain into another location of the body. So if you see somebody that in their head looks like really swollen, that has to do with hydrocephalus. It's dangerous because it's putting a lot of pressure on their brain, which is pushing on more nerves. And it's not safe. So does anybody have any questions before we go over to peripheral nervous system? We are almost out of this chapter, y'all. I'm rooting for it. We only got a few pages left. Anybody have any questions? Okay, well, I'm going to take just a second to check in with the group. All right. So if nobody has any questions, we're going to keep going to peripheral nervous system. So the nerves connecting the brain and spinal cord to other parts of the body constitute the peripheral nervous system. And this system includes cranial nerves and spinal nerves. So we talked about that earlier. There's the CNS and there's the PNS. And the PNS has two subdivisions, which is sympathetic and parasympathetic. So that connects the brain to the spinal cord, respectively, to the peripheral structures such as skin and skeletal muscles. So the ANS is considered a part of the PNS, and these connect the brain and spinal cords to various glands of the body, and to the cardiac and smooth muscles of the thoracic and abdomen. So then we go over to cranial nerves. There's 12 pairs of cranial nerves attached to the undersurface of the brain. So you can see these on page 193. Their fibers conduct impulses between brain and structures in the head and neck and the thoracic and abdominal cavities. So I'm not going to read the next part of that because it's going to be given to you in a chart on 194 that should be memorized before tomorrow. This is 9-3 on 194. So we need to know the 12 cranial nerves, what their conducted impulses are, and what their main functions are. So there's 31 pairs of nerves that are attached to the spinal cord in the following descending anatomical order. There's 8 pairs attached to the cervical segments, 12 are attached to the thoracic, 5 attached to the lumbar, and 5 attached to the sacral, and 1 attached to coccygeal segment. And this is over on figure 9-12. I want to see 9-12. I'll be pushing it here. Okay. So unlike cranial nerves, spinal nerves have no special names. Instead, they have a number that identify each one. So that's over on page 195. I'm not going to go over that because we don't have to know those. So I'm going to bump down just a little bit. Sometimes nerve fibers form several spinal nerves or reorganized to form a single peripheral nerve, and this mechanism can be seen as a network of interesting or braided branches called plexus. And 9-12 shows braided plexi. So the function of spinal nerves, they conduct impulses between spinal cord and body, different parts of the body that are not supplied by cranial nerves. Spinal nerves therefore function to make possible sensations and movements. A disease or injury that prevents conduction by a spinal nerve results in the loss of a feeling or loss of movement in the part supplied by the nerve. So detailed mapping of the skin surface reveals close relationship between the source of the spinal cord of each spinal nerve and the part of the body that innervates. So knowledge of segmental arrangement of spinal nerves is useful to physicians because we know if someone has an injury to a specific part of their brain or something is not able to be moved or done in the body, then we can usually associate that with which cranial nerve has been damaged. This helps us better to be able to care for our patients. So now we're going to move over to autonomic nervous system. It consists of 3 kinds of tissue. So we have cardiac muscle tissue, smooth muscle tissue, and glandular epithelial tissue, which we have gone over before in the past. And it's not going to be super, super serious that we know this, but I would at least have it in my brain. So the ANS includes parts of the nervous system that regulate involuntary functions, so heartbeat, contractions of the stomach, intestines, and secretions. It also has motor nerves that control voluntary actions of skeletal muscles or sometimes somatic nervous systems. The ANS consists of 2 divisions called parasympathetic and sympathetic. So autonomic neurons are the motor neurons that make up the ANS. The dendrites and cell bodies of some autonomic neurons are located in gray matter of the spinal cord or the brainstem, and their axons extend from those structures to terminate in peripheral junction boxes called ganglia. So these autonomic neurons are called preganglionic neurons because they conduct impulses between the spinal cord and the ganglion. All right, so in the autonomic ganglia, the axon endings of preganglionic neurons synapse with the dendrites or cell bodies of the preganglionic neurons. Postganglionic neurons, as their name suggests, conduct impulses from a ganglion to a cardiac muscle, smooth muscle, or glandular epithelial tissue. So autonomic or visceral effectors are the tissues to which autonomic neurons conduct impulses. Specifically, visceral effectors are cardiac muscle that makes up the wall of the heart, smooth muscle that partially makes up the walls of blood vessels and other hollow internal organs. A glandular epithelial tissue makes up the secreting part of the gland. So I don't think she's going to want us to know very much about that, but it's good to have it. All right, so autonomic conduction paths. Does anybody have any questions so far? We literally have like 2 pages left in this chapter, y'all. I may end up taking another break here in just a minute when we get at the end of this chapter before we go into the next one. If anybody has any questions, text them to me, group me to me, or comment. Okay. All right, so autonomic conduction paths. The conduction path to visceral and somatic effectors from the CNS, spinal cord or brainstem somewhat differ. So figure 9-20 is going to give us an example of those, and that is on page 197. So travel over preganglionic neurons from the spinal cord or brainstem to autonomic ganglia, where they're relayed across synapses to postganglionic neurons, which then conduct the impulses from the ganglia to visceral effectors. So compare an autonomic conduction path with a somatic conduction path illustrated on the left side in 9-20. We are going to bump over to sympathetic division. Sympathetic preganglionic neurons have dendrites and cell bodies in the gray matter of the thoracic and upper lumbar segments of the spinal cord. So for this reason, the sympathetic system has also been referred to as the thoracic-lumbar system. I don't think she's going to want to know very much about these. It leaves the spinal cord in the anterior ventral root of the spinal nerve, and it enters the spinal nerve but soon leaves it and branches to extend to sympathetic ganglion and through it to collateral ganglion. It synapses with several postganglionic neurons in the sympathetic and collateral ganglia, and a bump down to sympathetic postganglionic neurons. They have dendrites and cell bodies in sympathetic ganglia. So sympathetic ganglia are located in the front and each side of the spinal column. And they have a chain of two chains of beads that are often referred to as sympathetic chain ganglia. And like I said, this is on the top of 197 in this chart. So the functions of this, the sympathetic division functions as an emergency system. So we know that Ms. Putt said that the sympathetic, think of scared. So you've seen something, a snake, you're scared. This is going to be a protective function, like an emergency system. So impulses over some sympathetic fibers take control of many internal organs when we exercise strenuously. When we have strong emotions such as anger, fear, hate, anxiety, these are elicited. So in short, we must cope with the stress of any kind. Sympathetic impulses increase to many visceral effects and rapidly produce widespread changes in our body. So in the sympathetic division, when we've seen something that scares us, something that's made us uncomfortable, we are our bodies are built to go into a fight or flight mode. So your body's going to only take what it needs. So if you've seen a snake, you're not worried about digesting food. You are not worried about having to pee unless you accidentally pee on yourself. Like there's nothing that needs to be done except for getting away from that or fighting it, whichever you decide to do. So in this state, you are amped up, you're ready to go. Your body is just focusing on getting away or fighting what's coming at you. It's not concerned about the small stuff. It's looking at the bigger picture. So let's see. Sweat glands and adrenal glands secrete more abundantly. Salivary and other digestive glands secrete more sparingly. Digestive tract contractions or peristalsis become sluggish, hampering digestion. Like if something super serious happens to you, maybe like a car accident or something, it could be a while before you go to the bathroom after that because your peristalsis has been like, it's at a screeching halt because you are not concerned about going to the bathroom at that moment. You were concerned about whether or not you were going to live. So your body's going to be conducive to what's going on around you, if that makes sense. All right, so we're going to bump over to parasympathetic division. The dendrites and cell bodies of parasympathetic preganglion neurons are located in gray matter of the brainstem. Parasympathetic postganglion neurons lie within these outlying parasympathetic ganglia, and their short axons extend into the nearby structures. This is just the structure of it. We're going to bump down to functions. So parasympathetic system dominates control of many visceral effectors under normal everyday conditions. So this means you're going to have a normal heartbeat. You're going to have normal peristalsis. You're going to have normal functions, you can take a break, you can go to the restroom. Your eyes are not going to be dilated because there's nothing serious to see. Your eyes are going to be constricted because you're not trying to wonder who's coming at you, what's coming at you. So that's more of a normal. There's also a table 9-4 for autonomic functions on 198. I don't remember her saying that we needed to know this, but I would at least look at it so that you know what each of these visceral effectors are. So we're going to look at autonomic neurotransmitters. So in figure 9-21, the sympathetic preganglionic axon and parasympathetic preganglionic axon and parasympathetic postganglionic axon release acetylcholine, and these axons are therefore classified as cholergenic fibers. Only one type of autonomic axon releases the neurotransmitter norepinephrine, noradrenaline. This is the axon of a sympathetic postganglionic neuron, and such neurons are classified as adrenergic fibers. Adrenergic meaning like adrenaline. You have a hype of some type. So, okay. Each division of the ANS signals its effectors with a different neurotransmitter and explains how an organ can tell which division is stimulating it. So, for example, the heart responds to acetylcholine from a parasympathetic division by slowing down. On the other hand, the presence of norepinephrine at the heart is a signal from the sympathetic division, and the response is to increase the heart activity. So norepinephrine is going to bump you up. Acetylcholine is going to slow you down. Does anybody have any questions at this moment? I think a lot of the time we're just really overthinking this. Hope you guys are still hanging out. It's like 3 hours and 3.5 hours into this, but we're almost finished with this nervous system, and then we can go on. Okay. All right, so the function of the ANS is to regulate the body's autonomic involuntary functions in a way that maintain or quickly restore homeostasis. So the point is to bounce back as quickly as we can if something's gone wrong, if something has scared us or bothered us. Our body's just wanting to make sure that it's still living when it's over. Let's see. Many internal organs are dually innervated by the ANS. In other words, they receive fibers from the parasympathetic and sympathetic divisions. All right. So the term autonomic nervous system is something of a misnomer. It seems to imply this part of the nervous system is independent from other parts, but it's not true. Dendrites and cell bodies of preganglionic neurons are located in the spinal cord and brainstem, so they're continually influenced directly or indirectly by impulses of neurons located above them, notably by some of the hypothalamus and in the parts of cerebral cortex called the limbic system or emotional brain, which I feel like I'm in most of the time. So through conduction paths from these areas, emotions can produce widespread changes in the automatic functions of our bodies, in cardiac and smooth muscle contractions, and in secretion by glands. So, for example, anger and fear lead to an increased sympathetic activity or a fight or flight response, as we said. So according to some psychologists, the slightly altered state of consciousness is known as meditation, leads to decreased sympathetic activity in a group of changes opposite to those of the fight or flight response. So this is why people like to meditate, because it chills you out, it calms you down. All right, so that is the end of the nervous system, and I'm going to go back just a little bit, and I'm going to go through the boxes that they have. So any blue boxes or any extra stuff that we did not read through, I'm just going to go back and touch on that very lightly. All right, so let's go to 180 clinical application. It's a yellow box for multiple sclerosis. So many diseases are associated with disorders of the oligodendrocytes, which we read about. So these glial cells are involved in myelin formation, and these diseases are called myelin disorders. So a common primary disease in the CNS is a myelin disorder called multiple sclerosis, or MS. It's characterized by myelin loss and destruction accompanied by varying degrees of oligodendrocyte cell injury and death. So the result is a demyelination of white matter of the CNS, and its hard plaque-like lesions replace the destroyed myelin, and affected areas are invaded by inflammatory cells. Let's see, myelin Around axons is lost, nerve conduction is impaired, weakness, incoordination, visual impairment, and speech disturbances can occur. Although the disease occurs in both sexes, it's most common in women between 20 and 40. So the cause of MS is related to autoimmunity and viral infections in some individuals, and is typically chronic in nature, with remissions and relapses occurring over many years. However, some cases of acute and unremitting disease have been reported. There's no cure for MS. Early diagnosis and treatment can slow or stop its progression. All right, so now I'm going to go over to health and well-being on 183 for suppressing pain during exercise. So research shows that the release of endorphins increases during heavy exercise. Endorphins inhibit pain, so it's associated with muscle fatigue, and its fatigue decreases when endorphins are present, kind of like we talked about with ear piercings. So be sure and looking at these charts. Okay, now we're going to go over to Parkinson's disease on 187. This was also something that had to do with the muscular system before in chapter 8. But it's a chronic progression nervous disorder resulting from a deficiency of neurotransmitter dopamine in the basal nuclei of the cerebrum. So the group of signs associated with this disorder is Parkinsonism. Forward tilt of the trunk, you got reduced arm swinging, rigidity of trembling head, rigidity of trembling of extremities, shuffling gait, and short steps. So there's basically a gap between muscles and the nerves. Something's missing, a connection's not being made. So there's nothing wrong with this person's muscles necessarily. It's more on the nervous side. But there's people like, okay, Muhammad Ali, Michael J. Fox, they have this. So it's a misinformation in the part of the brain that normally prevents the skeletal muscle from being overstimulated. And it's a lack of dopamine, which is what keeps your body chill. So dopamine injection into the blood and dopamine pills are not effective treatments for Parkinson's disease because it cannot be sent over the blood-brain barrier, because the blood-brain barrier is intended to protect us from chemicals. Well, it's not going to let it in, and that's what's going to help with these patients. But there is something called levodopa or L-dopa, and it's found to increase the dopamine levels in afflicted patients. So neurons use it to cross the BBB to make dopamine, but for some reason it doesn't always work with a desired effect. And I'll go ahead and say something that's not in here, but surely it'll be in the next rendition of this book because it's moving along. But marijuana also helps patients with Parkinson's disease. It had, obviously, when you're high, it's going to increase dopamine, and that can pass the blood-brain barrier because it's usually through inhalation, or they'll put drops of this oil underneath your tongue. And within seconds, patients with Parkinson's disease begin to stop their tremors. And this doesn't last, for a very, very long time, but it does help. People with Parkinson's disease usually have to eat with like special utensils, bigger, handles on spoons and forks, because they can't hold it. They're shaking really, really hard. Also have found, just in my personal experience taking care of people in the nursing home that I know had Parkinson's disease, is being careful with their surroundings, because they're going to bump into stuff and involuntarily. They can't control that. So if you have a patient, you're making their bed, you may want to put something like I don't want to necessarily say a pillow, but something soft towards the rails of their beds so that they are not moving around, because I have seen patients that move involuntarily and have rubbed spaces onto their face because they can't help it if they're moving throughout the night and nobody's catching it. So the next thing that we're going to look at is, there was another bubble. Clinical application for lumbar puncture on 193, and this is talking about where a spinal tap or a lumbar puncture, which is also a posture that we had to memorize for checkoffs. This is when they put a needle just above or below the fourth lumbar vertebrae in the spinal cord, and okay, so this is have a patient on their side and arch their back by drawing their knees and chest together. It separates the vertebrae apart so that they can get into the space quicker. But since this spinal fluid is going throughout your body and into your brain, it's going to tell your body to rest when they give you, I'm trying to think of what the word is now. When you go in to have a baby and then they give you, What in the world am I trying to say? I can't think of the word. When you go in to have a baby and they give you the pain block, they put it in your spinal fluid and it sends a signal up to relax. So a lot of times women won't feel anything when they're having a baby, which can be a good thing. It can also be a bad thing. Miss McGee told us wanted to let us know that too much can cause a prolapse in your uterus. And I'm about to take a break just for a second so I can remember what this word was because we literally just talked about it. I think Lee sent it in the group. Y'all, we talk a lot in this group. I'm gonna go load it up. Okay. Okay. Pitocin. When you're being induced, pitocin. It can cause a prolapse in your uterus if there's too much because it's really, really, really contracting. Because we're trying to get that baby out, and if there's too much of that, it can cause your uterus to prolapse. All right. I think that's everything. No. 199. We have clinical application for herpes zoster or shingles. It's a unique viral infection that almost always affects the skin of a single dermatome. Nearly 15% of the population will suffer shingles at least once by the time they reach 80. It's itchy, it's irritating, it's not fun to have. It's usually around like your stomach area or shoulders. And let's see. So the disease results from a reactivation of the varicella virus, and the virus probably traveled through the cutaneous nerve and remained dormant in the dorsal root ganglion for years after an episode of chickenpox. All right, so that is everything for the nervous system, and we will come back in just a little bit. It's 6:36. Let's come back at like 6:45. We'll do 6:45 for senses chapter 10. All right, guys. So we are almost 4 hours into this. We're still kicking it. Thank y'all for hanging out with me. We can do this. It's a lot of information, but we are going to get through this. All right. So I'm going to go and roll on to the census chapter, which is the shortest, thank goodness. And it's really not that bad. This is more of a A&P kind of deal. I mean, she wants us to know what the functions are, obviously, because this will let us know later when we get to next semester. It's going to be important. Okay. All right, so we're going to start on page 209. This is chapter 10, census. All right, so let me just a second. All right. Sorry, we're still waiting on some people to enter into the group or try to get in. All right. So at the very top of this, it says there are millions of other sense organs throughout the body and our skin, internal organs, and muscles, and they constitute the many sensory receptors, which allow us to respond to stimuli such as touch, pressure, temperature, and pain. So our ability to detect changes in our external and internal environments is a requirement for maintaining homeostasis and survival in itself. So all these chapters are pulling together. We have to be able to understand that there's a stimulus, something's happening, something's changing, so that we can adapt with that, because if we don't, we won't survive a lot of the larger things in life. So there's a classification of senses. There's general and there's special. So these used to get me confused, because general you would think like plain Jane, normal, and then special you would think something crazy. but it's not like that. So general senses are detected by simple microscopic receptors that are distributed throughout the body and the skin, muscles, tendons, joints, and other internal organs of the body. They're responsible for sensations such as pain, temperature, touch, pressure, and body position. So, all right, let's get this going. All right, we're going to have general senses and special senses. We're going to go ahead and start with general. All right. So this is going to be responsible for pain, temp, touch, pressure, position. Okay, that's what that's responsible for. And then also it bumps down to special senses, a small area for that. These are detected by receptors that are grouped in specific areas associated with the complex structures that facilitate these senses. So smell, taste, vision, hearing, and equilibrium. And these are grouped with the distinct structures that enhance their function. So we have special senses, and this is smell, taste, touch, vision, hearing, and equilibrium. All right, so let's look at it like this. Generally, we're going to feel things. We're going to feel pain, temperature, touch, pressure, position. So generally, we're going to feel these. And then we are so special because we can smell, we can taste, we can touch, we can have vision, we have hearing, and we have equilibrium. So we are so special. Is how we'll remember that. And this is we generally can feel. I don't know if that helps, but that's how I'm going to try to remember that. So there's sensory receptor types, and these are not hard to remember because they are kind of a general, it's in their name. So we have photoreceptors. We have chemoreceptors. We have pain receptors. And we have thermo and mechano. All right. So photoreceptors is going to be color, light, vision. All right. Chemoreceptors is going to be taste, smell. All right. Pain receptors is going to be physical injury. All right. Thermoreceptors, temperature. And then the last one is going to be position or shape. All right. So then we're going to bump down to sensory pathways. And I actually have a sheet for this. I really hope that I printed that out because that was helpful to have. I'll be pretty sad if I don't have it. Actually, I think it might be on my phone. This is also on the website. I don't know if it's too late to print this out now. Let's see. It's in here. Okay, here we go. All right, so sensory pathways can be broken down. I'm going to go ahead and erase this because you guys can come back and look at that. All right. All right. So sensory pathways, I'm going to break it down into 4 steps. This is the best way that I know how to remember it. So 1, let's get that right. Sensory pathway. All right, so all sense organs must be able to detect a change or stimulus. All right. So we're going to say that this is our nose, and we are smelling something. This is the stimulus. All right. So we're going to go over to step 2. Detection of the stimulus must be converted to impulse. So stim converted to impulse. All right, so this is our nerve impulse, and then now we're going to go down to step 3. All right, so the signal might be conducted over a nervous system pathway in the brain. So signal conducted over system pathway to brain. All right, and so this is my nerve signal, and this is my brain. All right. All right, and so now we are at step 4, and then this is the brain perceiving the message. So your brain has to perceive the message. All right, I don't know if that helps. That's the way I remembered it. It's also on the website. There's a better picture of it on there, but that is the sensory pathway. So that is the what has to take place for a senses to even happen. So, yeah. All right. So general sense is the distribution of general sense receptors. So microscopic general sense organs receptors are found in almost every part of the body, but they're not concentrated in the skin. These receptors are not evenly distributed over the body surface or in the internal organs. They all do not all respond to the same type of stimulus. So each thing does a different thing. It's structure fits function. Structure fits function. It's going to be in everything that we read. All right. So, all right. So examples of general sensory receptors and their modes is going to be in table 10-1. So you have free nerve endings, and this is a naked nerve ending, and there's several of those. So it's in skin and mucosa, epithelial layers, pain, descriptive, touch, tickle, temperature. There's encapsulated nerve endings. There's bulboid corpuscle, which is in the subcutaneous tissue, mucosa of the lips and eyelids, and external genitalia. And this is touch and possibly cold. Lamellar corpuscle or Pacini corpuscle, we talked about that before, in the subcutaneous and subserous tissue around joints and mammary glands and external genitalias in both sexes. So this is a pressure and high frequency vibration. Tactile corpuscle or Meissner corpuscle, we did talk about that before. It's in the skin of the dermis, fingertips, and lips. And there's fine touch and low frequency vibration. Bulbous corpuscle or Ruffini is the skin, dermal layer, subcutaneous tissue of fingers. It involves touch and pressure. Tendon organ or Golgi tendon organ is near the junction of tendons and muscles, and its proprioception sense of muscle tension. And then you have muscle spindle, which is in your skeletal muscles, and its proprioception sense of muscle length. We also need to look down at table 10-2 for the special sense organs. So those two are going to break those down for you. All right, so the ability to distinguish one touch stimulus from two is called a two-point discrimination, and there's a neurological test for this. It doesn't tell you what it's called, but it basically tells you if you touch one part of a person's body, they should be able to, touch two parts of a person's body, they should be able to tell you the difference. This was in a Jessica Niwakausha video. I can't remember how to say her name. She did great. This was one that Mr. Vi told us about, and it wasn't something that we had to know for physical assessment, but this was something that she had talked about. So modes of sensation are the stimulation of general sensory receptors can lead to a variety of sensations, and the difference in which kind of stimuli is detected is called the mode of sensation. So different general sensory receptors can detect vibration, deep pressure, and light pressure, pain, stretch, or temperature. So some general sensory receptors are found near the point of junction between tendons and muscles, and others are found deep within skeletal muscle tissue, and they're called proprioceptors. So when they're stimulated by a stretch, these mechanoreceptors provide us with information concerning the position or movement of the different parts of the body, as well as the length and extent of contraction over our muscles. Golgi tendon or Golgi tendon organ and muscle spindles are also proprioceptors. Many general sensory receptors are found in the skin, but some are present deep in the body. So there's stretch receptors in your stomach that signal when you're full. There's stretch receptors in most other organs like your intestines, arteries, vagina, birth canal, urinary bladder, and they enable the normal functions of those organs. So there's also an important chemoreceptor in the aorta and other arteries that detect changes in pH and carbon dioxide or CO2 levels in the blood, and important information for regulating breathing and heart rate. So now we're going to go down to special senses at the bottom 211. Vision detects the color and intensity of light in our external environment, but when focused by the eyes and processed by the brain, it can do more. So we can recognize outlines of depths of objects, analyze movement, and determine distances. So there is layers of the eyeball. We're going to get into the structure and function of the eyeball on 212. You have a fibrous layer, you have a vascular layer, and you have an inner layer. So in the fibrous, you have the sclera and cornea. Vascular, you have choroid, ciliary muscles, iris, and lens. And then inner layer, you have a retina, optic nerve, and retinal blood vessels. So in the fibrous layer, it's a tough fibrous tissue. The white part is your sclera. This white part in your eye is your sclera. The dense bundles of collagen forms most of this. There's a transparent circle in the anterior of the fibrous layer called the cornea. So if you're thinking of your eyeball, these are the layers around this. So you have this layer is the white, then you have a choroid, and then you have a retina, which is the very, very back portion of it. So there's a mucous membrane. I'm sorry, let's back up. Inflammation of the cornea is keratitis, which she might throw that in there. Any change in shape of the cornea can dramatically change the ability of the eye to focus on the retina. Okay, mucous membrane is known as a conjunctiva. So when we pull down our patient's eyes to look at our conjunctiva, we want to make sure that they're pink. If they're white, that means they're dehydrated. Conjunctiva that's inflamed is conjunctivitis. So itis is inflammation. So conjunctivitis is pink eye. Pink eye is a infection of the conjunctiva, and it's called conjunctivitis. Also, we have a lacrimal gland in here, which if you look really, really close at your own eye in the mirror, there's a teeny tiny little hole in the corner right here on each of your eyes. That's your lacrimal duct or lacrimal gland. And this is what secretes your tears. This is where your tears come from. If you ever cried in front of a mirror, maybe, you can see them come from that. So this just keeps your eye moist. This is what keeps your eyes from drying out. All right, so we have a vascular layer. It's the middle layer of the eyeball, and it's a dense network of blood vessels. So this is made up of the choroid, which contains a large amount of the dark pigment melanin, and it's almost a black layer that helps prevent scattering of incoming light rays. So several involuntary muscles make up the anterior part of the choroid. Some are the iris, and the iris is the colored part of your eyeball. So this is going to be the, I'm sorry. Yeah. It's the colored part. So this is what I have brown eyes. Iris is the colored part. So then you have the black center, that's the pupil, and this is what constricts so that you can either let more light in or you can let less light in. So if I'm looking at this light up here, my eyes are going to kind of constrict because it's too much light and it's going to not be good for my eyes. So they're going to kind of kick that out a little bit. And if I'm in the dark, my eyes are going to dilate because I want to see as best as I can at night. That's why sometimes when you first turn the light off at night to go to bed, it's completely black because your eyes have not dilated back out yet so that you can kind of see in what's called your night vision. So the thing that controls, you have the lens, which is directly behind the pupil. So the pupil is kind of like a donut. It's just a hole in the middle, if that makes any sense. Then you have your lens, which is behind it, and this is held in place by ciliary muscles, which are just like muscles on each side of it, and they are going to help to constrict and dilate your pupil. So let's see. When the ciliary muscle is relaxed, the lens has a slightly curved shape to focus on near objects. The ciliary muscle must contract. As it contracts, it pulls the choroid forward towards the lens, and it causes the lens to bulge and curve even more. So most of us are become farsighted as we grow older. Our lenses lose at least some of its elasticity and can no longer bulge. And this is called presbyopia is called old-sightedness, and it's a name for this condition. So most young people lens have both transparent and somewhat elastic capabilities. And now we're going to go over to page 214. So it says, unfortunately, in some individuals, long-time exposure to ultraviolet radiation and sunlight can cause the lens to become hard and lose transparency, and they kind of become milky in appearance, and this is called a cataract, and you can have a surgery to correct this. But the defective lenses are replaced with an artificial implant. So the inner layer, the retina, makes up most of the inner layer of the eyeball, and microscopic photoreceptor cells are there to detect light. So most of them are called rods and cones. So rods are going to be your night vision, and cones are going to be your daytime, and they're going to be responsible for color. So the three kinds of cones, which are sensitive to different color, red, green, or blue, are scattered throughout the central portion of the retina, and these are allowed us to distinguish between different colors, but only in bright light. So we wouldn't be able to do that at night. There's a yellowish area near the center of the retina called the macula lutea, which means a yellow spot, and it surrounds a small depression in the fovea centralis. Okay. And that's where the greatest concentration of cones of any area are in the retina. So you can look at these with an ophthalmoscope. That's what they use when you go to the doctor's office. They want to look at your eyeballs. So there's a figure 10-4 that shows ganglion cells, which are sensitive to light. They are like rods. They're sensitive to various wavelengths or colors, but they're not used to form visual images. They're used to help the body determine whether it's day or night and whether the level of moonlight or monthly phases. It helps our body's internal clock mechanisms synchronize themselves into daily, monthly, and seasonal rhythms for our internal environment. So there's fluid in your eyeball, and you have to have this, and you can have too much and you can have too little. So the fluid that fills the hollow spaces in the eyeball helps maintain the shape and reflect light rays. So fluid bends light rays to bring them into focus of the retina. And sometimes when I think about that, I think of water. If you like put something behind it and then you look through a clear glass, it kind of flips it, which is kind of what happens in our brain. So aqueous humor is the watery fluid at the front in the anterior chamber, and visceral fluid is the thicker one in the posterior chamber in the back of your eye. So I think A comes first in the alphabet, it's going to be in the front. V comes later in the alphabet, it's going to be at the back. So if you have too much, if drainage is blocked for any reason in the internal pressure within the eye, it's going to increase, and damage is going to lead to blindness, and this condition is called glaucoma. So we have visual pathways. Light is stimulated that results in vision. That's our ability to see the objects that exist in our environment. Light enters through the eye, through the pupil, and is refracted or bent. So like we were talking about the glass. Refraction occurs as light passes through the cornea and the aqueous humor and the lens and vitreous humor on its way to the retina. So the innermost layer of the contains rods and cones, which are the photoreceptor cells. This has to do with vision. All right, nerve signals eventually leave the retina and exit the eye through the optic nerve, which is cranial nerve 2, and the posterior surface of the eyeball. No rods or cones are present in that area of the retina, and the result is a blind spot known as the optic disc. So after leaving the eye, optic nerve enters the brain and travels to the visual cortex of the occipital lobe of the cerebrum, and eventually visual interpretation of the nervous impulse is generated by light striking the retina and causing us to see. So let's take a little break. We're on page 217. Yeah, we only got just a little bit more after this. We're going to take a break, and then we're going to come back and start on hearing and equilibrium. So let's come back at 7:15. All right, so I'm going to pick back up at page 217 under hearing and equilibrium. So in addition to its role in hearing, the ear also functions in the sense organ of balance or equilibrium. So stimulation or trigger that activates receptors involved with hearing and equilibrium is mechanical, and those receptors are called mechanoreceptors. Mechanoreceptors. Oh, somebody was calling. Okay. All right, so in hearing, sound vibrations trigger nervous impulses that are eventually perceived in the brain as sound. And equilibrium changes in position or movement of the body trigger impulses that lead to the sensations of balance. So there's structures in your ear, and Callie just sent something to the group a little while ago that was very helpful. So if you want to remember these in order, you have the external ear, the middle ear, and the inner ear. I'm sorry. The thing she sent is going to be just a little bit later. It has to do with the middle ear. But all right, so the external ear has two parts. You have the auricle or the pinna, which is this part of your ear that we had to pull back when we did physical assessment, and the external acoustic canal. This auricle is the appendage on the side of the head surrounding the opening of the external acoustic canal. So the canal itself is a curved tubing, and you can see that on page 218 in that figure 10-6. And it extends into the temporal bone at the tympanic membrane or the eardrum. So the tympanic membrane, another word for that is eardrum. The skin of the acoustic canal contains short hairs and ceruminous glands, which form cerumen, which is ear wax. So it keeps the canal from drying out, and excess cerumen in the canal can impair hearing by absorbing or blocking the passage of sound waves. So we want to make sure that we're cleaning our ears, but that we're not cleaning them too much and drying them out, because it does need to be lubricated. So then if you go to the doctor and they need to look at your ear, they're going to use an otoscope, and this is how they look to see if there's any inflammation on your eardrum or anything else going on inside of your ear. So we're going to bump over to middle ear on page 219. So you have ossicles, malleus, incus, and stapes. Okay, so the middle ear is a tiny and very thin epithelium-lined cavity hollowed out with the temporal bone, and there's three very small bones. So all of them collectively are ossicles, and you have malleus, which is your hammer, incus, which is the anvil, and stapes, which is your stirrup. So the handle of the malleus attaches to the inside of the tympanic membrane, and the head attaches to the incus attaches to the stapes, and this covers a small opening of the oval window. So you're going to have to look at 10-8. To better understand that, and 10-6. So it is important to know that the auditory tube or station tube connects to the throat with the middle ear. So that's why a lot of the times when we have a sore throat, it might spread to produce a middle ear infection called otis media. So an ear infection is otis media. Function between healthy auditory tube is to equalize air pressure between the middle ear and the outside environment. And when air pressure is unequal, the tympanic membrane may remain stretched, sometimes becoming quite painful, reducing its ability to vibrate, so reducing our ability to hear things well. So the inner ear consists of three spaces. We have a bony labyrinth, and it's odd-shaped bony space filled with watery fluid called perilymph, and it's divided into the parts called a vestibule, semicircular canal, and cochlea. So the vestibule is adjacent to the oval window between the semicircular canals and the cochlea. And we're not really going to need it. She's not making us label anything. We just need to know what these parts are. So the balloon-like membranous sac is suspended in the perilymph and follows the shape of the bony labyrinth, which is a tube within a tube. And this is the membranous labyrinth, and it's filled with a thicker fluid called endolymph. So now we're going to go to hearing on 219. It's a sensation of both intensity or loudness, frequency, which is tone, of sounds in our environment. So sound waves are simply pressure waves in the air. They can be funneled by the auricle to the external acoustic wave canal and strike the tympanic membrane. So it's going to hit that. It's almost like a trampoline, I kind of think of it, and it's going to bounce off of that. So that movement is transmitted and amplified inside the ear ossicles as it passes through the middle ear. And the movement of the stapes against the oval window causes the movement of perilymph fluid through the inner canal, which in turn triggers vibrations of the endolymph. So the spinal organ or organ of Corti is the organ of hearing. And then you also have a cochlear nerve. So the activation of mechanoreceptors in the spiral organ inside the cochlea of the inner ear generates nervous impulses that travel through the cochlear nerve to the brain. So the organ of hearing is the organ of corti, and then the cochlea is like the main part inside of that organ. So if you have somebody who has hearing issues, they can have a cochlear implant, and this is to help them hear. So we have presbycusis, which is a hearing loss. It's progressive hearing loss associated with aging, results from degeneration of sensory nerve tissue in the ear and vestibulocochlear nerve. So let's see, different sound frequencies, tones stimulate different regions of the spinal organ, therefore, or spiral organ. Hearing impairment is limited to only frequencies associated with the damaged portion of the spiral organ. So this has to do with the ciliated, the little hairs that are inside. Different parts of those hair pick up different frequencies. So if you have damage to certain parts of those, you won't be able to hear, whether it's like high tones or low tones, you won't be able to hear those. So we're going to go over to equilibrium. So mechanoreceptors for these are sense of balance. The equilibrium is located in a sac-like vestibule in the 3 semicircular canals of the inner ear. So I'm going to go over to hearing and equilibrium in my study guide. All right. Let's see. All right. So 2 types of balance are static and dynamic. Give me just a second. Somebody just sent me a message. Yes. Okay. All right. So each of the structures that are these vestibules are made up of a patch of sensory hairs coated in a thick glob of heavy gel. And so this also has to do with the hairs inside of your ear. These are called macula. So when you bend your head, gravity acts on the heavy gel to pull it one way or the other. And this in turn bends the cilia of the hair and produces a nerve signal. So the nerve is interpreted in our brain as a sense of gravity or static equilibrium. 3 semicircular canals are half circles oriented at right angles to one another, which each canal, or within each canal, is the dilated area called ampulla that contains a sensory structure called crista ampullaris, which generates a nerve impulse when you speed in a direction or movement of your head changes. This sense of motion is called dynamic equilibrium. So when I think of static equilibrium, I'm bending over a little bit, and it's going to let my body know that there's gravity in play, and it's going to keep me from just keep going over and falling out. So then dynamic is a sense of motion. So as I'm spinning in this chair, this is keeping me from flying out, falling out, freaking out, because it's pulling on those hairs, and it's letting my body know that there's a sense of motion, that I'm moving. So one is keeping me from losing my balance, and the other is letting me know that, okay, she's spinning, okay, she's moving. These are in figure 10-10 and figure 10-11. So sensory cells in the cristae ampullaris have hair-like cilia that are embedded in flap-like cupula, which sways back and forth within the indolence. Sensory cells are stimulated with a change in environment of the head, causes indolence to move differently, causing the crista ampullaris to sway more with more or less force. So nerves from mechanoreceptors in the vestibule join those from the semicircular canals to form a vestibular nerve, vestibulocochlear nerve, cranial nerve. This is all on page 221. These are going to be also mentioned in the nervous system chapter. All right, so we're going to go over to taste at the bottom of 221. So our sense of taste is gustation and allows us to chemically analyze the food before we bite or swallow it. So when you smell something, this is gustation. Well, gustation is tasting it, but you're going to smell that, and you're going to kind of know what it tastes like before you taste it, if that makes any sense. And that's just chemically analyzing in our brain. So the taste buds are where you're going to taste your food at. They contain supporting cells and chemoreceptors called gustatory cells. These generate nervous impulses to the brain as taste. These bumps are called papillae. So your taste buds are called papillae, and the large ones at the inverted V pattern at the back of your tongue are circumvallate papillae. So chemicals dissolved in the saliva stimulate the chemoreceptor gustatory hairs. Nervous impulses that are generated by stimulation of taste buds travel primarily through 2 cranial nerves to end in the taste area of the cerebral cortex. So there's 4 primary taste sensations, and it's sweet, sour, bitter, and salty. And you have 2 more, and it's metallic to taste to detect metal ions, and then savory, meaty taste, which is umami, and that's to detect amino acid glutamate. So those have been added to the primary list, but there's 4, and then there's 2 little extra ones on the side. So let's see. Now we're going to go down to smell. So the smell, sense of smell or olfaction helps us to detect certain chemicals in our environment. Chemoreceptors are responsible for olfaction, and they're located in the epithelial tissue in the upper part of the nasal cavity. The location of olfactory, which is smell, receptors is somewhat hidden, and we can often forcefully sniff the air to smell things that we want to smell. So when I go through the candle aisle at Walmart, I'm going to use my olfactory receptors to intentionally smell things. So each olfactory cell has a number of sensory cilia that detect different chemicals and cause the cell to respond by generating a nerve impulse. So they're extremely sensitive and respond quickly to even slight odors. So after a short amount of time, they develop a kind of fatigue and lose their ability to respond. They decrease in receptor sensitivity, and this is called adaptation. So that's why when, like right now, my husband's making fish downstairs, and it's like super strong and I can smell it. But after a while, I'm not gonna be able to smell it anymore because I'm gonna be adapting to this, and it's just gonna be something that's diffused in the air around me. So after these are stimulated by odor-causing chemicals, the resulting nerve impulse travels through the olfactory neurons in the olfactory bulb and cranial nerve 1, and then enters the thalamic and olfactory centers of the brain, where the nervous impulses are interpreted into specific odors. So pathways taken by these nerve impulses and areas are interpreted closely and associated with the limbic system of the brain, which we talked about the limbic system of the brain before. I don't know how far back that was and what chapter it was, but I do know we talked about it. And it was a really small part. I really want to find it now. It had to do with this. Yeah. Okay, so on page 199, let's see, the limbic system or emotional brain. So this has to do with the autonomic nervous system. All right, so, and that also ties into smell as well. So it's important in memory and emotion, which is exactly what it said on the other page. So that's why you can smell something and think of something in the past that has been associated with that smell. So the smell of bread or cookies baking in a grandmother's kitchen may be a part of a childhood memory that lasts a lifetime. You're never going to forget that smell. All right, so integration of senses. So this is a big picture of sensation. We should remind ourselves that they're perceived in the brain. Not all individual receptors scattered throughout the brain. Some are, they never get to the brain, and others are amplified or muffled in the brain. So some foods seem to have a different flavor if they're crispy, warm, or cold. Some spicy stimulate pain or temperature receptors that produce a hot flavor. Smell sensations, even more than others, trigger memories. We often combine senses of equilibrium and vision and proprioception to maintain our posture and balance. We also need to be reminded that our senses do fail us as we age. Those receptors just aren't as strong as they once were. So it says this may explain why some foods just don't taste the same as they did when you were younger. So that's all of chapter 10, senses. Now we're going to go over to chapter 11, endocrine system. And this is the long one, but it shouldn't take us that long because we're just going to bump through this. I'm going to go back real quick. Let me bookmark this. I'm going to go back real quick and make sure we didn't miss any bubbles in chapter 10. These like clinical applications and stuff, we might need to know those. So I like to go back and look. We have tables. All right, so we have referred pain. This is a stimulation of pain receptors in deep structures may be felt in the pain as pain in the skin that lies over the affected organ or the area of the skin or body surface removed far from the site of disease. And this is referred pain. So when you say like, let's see. Okay, classic example is a patient having a heart attack. So you could say like your chest hurts. Let's see. trying to find a way to explain this. I know she did. So, for example, pain originating in a deep organ of the abdominal cavity is often interpreted coming from an area of skin whose sensory fibers enter the same segment of the spinal cord. So, when I say that I have sorry. Okay, I'm coming.
You want me to bring it up?
No, I'm good. So referred pain would be when you said maybe you have a stomach ache and it's not necessarily your stomach, it's just an area around it, but you're referring to something in that region. All right, so clinical application is macular degeneration on page 215. So age-related macular degeneration is for people over 65, and then the most common type is called dry AMD. And so macular area of the retina degenerates in a disease process over time, and central vision field is gradually lost, and the ability to distinguish fine detail diminishes. And then there's also an application on page 214 for visual acuity. And this is the clearness or sharpness of visual perception, and acuity is affected by our focusing ability, the efficiency of the retina, and the proper function of visual pathway and processing centers in the brain. There's a place for you to find your blind spot. There is, you definitely need to know myopia and hyperopia. So myopia is nearsightedness and hyperopia is farsightedness. So myopia would say, I'm so nearsighted. And then hyperopia is farsighted. Let's see. All right, we have photopigments in the cones. Color blindness is due to a mistake in producing three chemicals, which are photopigments in cones. Each photopigment is sensitive to one of three primary colors, and that's blue, green, and red. I would know that. Okay, we have external otitis, which is swimmer's ear, and it can be bacterial or fungal, and it's in the auditory canal and auricle. So that's when you just get too much water in your ear. Then we also have, I think there's one more. Maybe not. Nope, that's it. All right, so now we're going to go to chapter 11, the endocrine system. So if you guys want to take a little break, I'm going to get my papers together over here. Valerie said, ooh, fish. Yeah, he just came up here and asked me if I wanted some. I'm like, I'm good. I have my energy drink. I don't think I'm going to eat till tomorrow. So you guys go ahead and take a break if you want to. I'm going to go ahead and get my papers together and get ready for this one. And this is the last stretch. And then you guys can do y'all's thing. All right, I'm ready to start if you guys are. Let's see. All right, so let's go ahead and get started. Endocrine system and see if we can blast through this. So the main functions of this are communication and control. Endocrine glands Let's just make this real easy. Marker. All right. All right, so exocrine glands exit through ducts, and then endocrine glands go into the blood. All right. All right. So endocrine glands are ductless, and they secrete hormone or chemicals known as hormones. Go ahead and do this. All right. So each hormone molecule can bind to a cell that has a specific receptor for that hormone, triggering a reaction in the cell. And these cells are called target cells. So hormones are going to be secreted, and they're going to attach to target cells. Okay. All right. So if there's too much of a gland that's secreted, too much of a hormone that's secreted, this is hypersecretion, and too little is hyposecretion. So too much, hyper, too little, hypo. Hypo. All right, so moving right along, mechanisms of a hormone action. So you have non-steroid and steroidal. All right, so major classes of hormones. They differ in mechanisms by which they influence target cells. And it's important to understand a few characteristics of these. So we're going to start with non-steroid hormones. These are whole proteins in shorter chains of amino acids, simple versions of single amino acids. Non-steroid hormones typically work according to second messenger mechanism. So according to this concept, a non-steroid protein hormone, such as thyroid-stimulating hormone, acts as a first messenger. It delivers its chemical message from the cells of the endocrine gland to highly specific membrane receptors in target cells. And the interaction between the hormone and specific receptor site and the plasma membrane is often like a lock and key mechanism. So after a hormone attaches to a specific receptor site, number of chemical reactions occurs, and the reaction activate molecules within a cell called second messengers. So the way that I'm going to do this is, and I don't know if she wants us to know it this deep, but I just like to see it in my mind. So we're going to do it like that. We have a non-steroid hormone, and it has been secreted. It's a whole protein. All right. All right. Non-steroid protein hormone is the first messenger. So this hormone, the non-steroid hormone, is the first messenger. And it is going to, it delivers its chemical message from the cells of the endocrine gland to highly specific membrane receptors on target cells. So the interaction between the hormone and specific receptor is the lock and key. So when it attaches to the specific receptor site, a number of reactions occur, and the reactions activate molecules within the cell called second messenger. So when it attaches to receptor, then it is a second messenger. Okay. All right, let's see. One example of this mechanism occurs when the hormone receptor and interaction changes energy-rich ATP molecules with inside the cell to cyclic AMP or adenosine monophosphate. Cyclic AMP serves as the second messenger delivering information inside the cell that regulates the cell's activity. So cyclic AMP causes thyroid cells to respond to thyroid-stimulating hormone by secreting a thyroid hormone such as thyroxine. Cyclic AMP is only one of several second messengers that have been discovered. So I'm not going to get too deep into that. I would know the golden terms for that, but this is basically how that works. Okay, no. The table 11-1 on 233. This is literally about to be your life through that entire test. I don't know if she's going to do matching with this or she's going to do multiple choice, but that's basically this entire chapter in chart form. So we're going to go past that. We're going to look at steroid hormones. So primary actions of a small lipid-soluble steroid hormone such as estrogen do not occur by a second messenger system because they are lipid-soluble. Steroid hormones can pass intact directly through the plasma membrane of the target cell. So once inside the cell, steroid hormones pass through the cytoplasm and enter the nucleus where they bind with a receptor and they form a hormone receptor complex. So this complex acts on DNA and it ultimately causes the formation of a new protein in the cytoplasm and then produces specific element or effects in the target cell. So in the case of estrogen, it might be breast development in female adolescent. All right, so because it takes time to accomplish all these, yes. So we're down to that one. They're slower. Besides the primary effects of steroid produced by the DNA triggering mechanism, steroid hormones may also trigger membrane receptors to produce a variety of secondary effects. So the secondary effects usually appear more rapidly than they do primary steroid effects. So then we're going to go over to 235 for regulation of hormone secretion. This is negative feedback and positive feedback. We talked about this before. We're going to say regulation of hormone levels in the blood depends primarily on the homeostatic mechanism called negative feedback. So negative feedback, let me see if I can find a better definition for this. It says when released, hormone insulin is an example. When released from endocrine cells in the pancreas, insulin lowers blood sugar levels or glucose concentration in the blood. Normally, elevated blood glucose levels occur after a meal, after the absorption of sugars in the digestion tract takes place. Elevated blood glucose stimulates the release of insulin from the pancreas. Insulin then assists the transfer from sugar to the blood into cells, causing blood glucose levels to drop back towards the normal set point. So as blood glucose concentration levels drop, the endocrine cells in the pancreas slow their production and release insulin. These receptors are negative because the reverse direction of disturbance to the stability of internal environments of the body. So the way that I remember this, and you should be able to tie the two together, is a negative is going to be something that's working oppositely of what's going on. So it wants to do the opposite effect. If your blood sugar is raised, it's going to try to find a way to lower it. If it's low, it's going to try to find a way to raise it. It's working in the opposite direction. So we're going to put negative, reverse, or opposite. All right, and then for positive, we're all for it. All right, so positive, the way that I remember this was how it was explained in the other, and it's actually right here, muscle contractions to push a baby through the birth canal. They become stronger because positive feedback is going to encourage that. It's wanting to secrete the hormone oxytocin so that this baby can be pushed out. So it's going to work towards that. All right, so there's levels of regulation. Let's see. Typically, additional levels of control are involved in maintaining homeostasis. Feedback may trigger autonomic nervous stimulations of a gland and then secrete a releasing hormone. Often, all levels of control are receiving and reacting to feedback. So we're going to bump down to prostaglandins. PGs, or tissue hormones, are important and extremely powerful lipid substances found in a wide variety of tissues. They're modified versions of fatty acids and play an important role in communication and in the control of many body functions, but do not meet the definition of a typical hormone. I don't think we're going to need to know much about that. So PGs, prostaglandins are, along with several other tissue hormones such as leukotrienes, thromboxane, and there are sometimes paracrine agents. The term paracrine means secrete beside, and it's a description for regulatory agent release right next to its target cell. Let's see, so you have prostaglandin A, prostaglandin E, prostaglandin F, and they're the best known. They have profound effects on the body functions. They influence respiration, blood pressure, gastrointestinal secretions, inflammation, and reproductive system. They believe that they regulate cells by influencing the production of cyclic AMP. So let's see. So in fact, many common treatments such as aspirin exert their effects by altering the functions of PGs in the body, so next to where it's being released. So we're going to look at the pituitary gland. It's also called an adenohypophysis, and the pituitary gland is the neurohypophysis. I'm not going to be very deep into that because you can read this on the chart, and I'll just go back and do that. So basically, your pituitary gland sits in a sella turcica, which is the seat formed by two bony projections in the sphenoid bone. This is just basically put safely in your brain so that nothing happens to it. So you have an anterior pituitary hormone, and the four hormones that are inside of it are tropic hormones. And they stimulate another endocrine gland to grow and secrete its hormones. So you have thyroid stimulating hormone, adrenocorticotropic hormone, follicle stimulating hormone, luteinizing hormone, growth hormone, prolactin, and then we move on to the next. So I'm going to go through and pick out the keywords, and then we're going to go back and look at the chart. So let's see. All right, so anabolism is a buildup of an amino acid. We do need to know that to form tissue proteins. And then too much insulin in the blood is hypoglycemia, and too little is, too much insulin in the blood is hypoglycemia, and then too much GH produces hyperglycemia, which is a higher than normal blood glucose concentration. So I would know those are on page 237. Also called human growth hormone, is sometimes used by people to keep themselves youthful or to boost athletic performance. We also need to know this box, the clinical application on 237. So giantism is a hypersecretion of a growth hormone. So growth hormone is pretty self-explanatory. If you have too much of it, you're going to be huge. So there's a picture of that here. And then you have dwarfism, which is a hyposecretion of a growth hormone. If there's too little of it, dwarfism would be a effect of that. So then we also have, if you have too much of if the pituitary gland secretes too much growth hormone after normal growth years, then the disease is called acromegaly. And this is an enlargement of bones in the hands, feet, jaws, and face. And then there's a picture of that there as well. You can also YouTube or Google that kind of stuff. It's pretty cool. All right, so let's go back to the chart on page 233, and we'll go through the anterior pituitary glands real quick. So the pituitary gland is the master gland because it's got so much going on inside of it. It's basically going to run the show. So you have TSH, or thyroid stimulating hormone. So this stimulates the secretion of thyroid hormones, which is down later in the box. So basically this is, it's going to work to help another part work. So they're kind of partners, if you want to look at it that way. So then we have a adrenocorticotropic hormone, or ACTH. This stimulates the secretion of the adrenal cortex hormones, which is over in your glands above your, it's above your kidneys, adrenal glands. Yes. All right, so follicle stimulating hormone, or FSH, in females, it's going to stimulate development of ovarian follicles and secretion of estrogens. In a male, it's going to stimulate seminiferous tubules of testes to grow and produce sperm. So this is your body getting ready. So follicle stimulating, it's going to stimulate something to get started. So you're going to start developing ovarian follicles and the secretion of estrogen. So this would be when you are getting ready, probably, to start your cycle. And then males, this is when you're hitting puberty. It's going to stimulate for you to start producing sperm. All right, so then we're going to go down to luteinizing hormone, which is LH. It's going to stimulate maturation of ovarian follicle and ova, stimulates secretion of estrogen, triggers ovulation, stimulates development of corpus luteum, or luteinization, which is going to be talked about later in the reproductive chapter. And this is basically the steps that it takes for you to get pregnant. So this is when you are having your cycle. This is during puberty. So male stimulates interstitial cells of the testes to secrete testosterone. This would also be puberty. Growth hormone, GH, stimulates growth in all organs, metabolizes nutrients, molecules, causing an increase in blood glucose concentration. So this is where we talked about acromegaly, dwarfism, things like that. So prolactin, or PRL, is a lactogenic hormone, and it stimulates breast development during pregnancy and milk secretion after pregnancy, so breastfeeding. So now we're going to go back to posterior pituitary gland hormones. She said to know this figure 11-5 on 238, so I would definitely know that. So neurohypophysis releases two hormones, only two, and it is the antidiuretic hormone and oxytocin. So antidiuretic hormone is what is going to control like water balance, fluid electrolyte balance in your kidneys. So it's very important to have this. So if you're taking, I'm trying to think of what the word is, the medicine for this. I can't think of it. I'll have to get back to it, but there's a medicine that you take that causes you to have over or under of it. So hypersecretion or hyposecretion of ADH results in diabetes insipidus, a condition in which large volumes of urine are formed. Dehydration and electrolyte imbalances may cause serious problems. And then we have oxytocin, which is secreted high levels by a woman's body before and after she has a baby. It stimulates contraction of smooth muscle in the uterus. And that's believed to initiate and maintain labor. This is why they prescribe oxytocin to inject, to induce ADH acts to decrease urine volume and prevent dehydration. Simplest way of saying it. Okay, thank you, Valerie. All right, so she said ADH acts to decrease urine volume and prevent dehydration. So that's the simplest way to memorize that. So back to oxytocin. This is basically having to do with a female milk letdown. This has to do with contractions, and this is also why if you're going to be induced to have a baby, they put this into your spinal cord or spinal fluid, and this is what allows that to be sped up. So, but this is also what we talked about, where you can get too much of it, and it can cause a prolapse in your uterus because it's contracting so heavy. So then we're going to bump down to hypothalamus. All right. So this one has a lot going on. All right, so these hormones are released from the posterior lobe of the pituitary, and actual production of these two hormones occurs in the hypothalamus. So two groups of secretory neurons in the hypothalamus synthesize the posterior pituitary hormones, and then they release, they pass along axons to the pituitary gland. So the release of ADH and oxytocin to the blood is controlled by the nervous stimulation. So it's going to say that there's only two in this box on page 233, but it's also got to do with oxytocin and ADH because they come from that gland down here. All right, so it also produces releasing hormones and inhibiting hormones. So these are produced in the hypothalamus. I'm going to go back to this chart. So releasing hormones stimulate the anterior pituitary to release hormones. And then inhibiting hormones are going to inhibit the anterior pituitary secretion of the hormones. So one is stimulating and one is inhibiting. Give me just a second. Okay. So we're going to bump over to thyroid gland. So they're organized into different chambers called thyroid follicles. And so these are, how to picture them better. And I don't know what I did with it. But so just know that they're the chambers in the thyroid gland. So each thyroid follicle is filled with a thick fluid called colloid, which contains many fine suspended particles that store thyroid hormones. So the thyroid gland secretes two thyroid hormones and the hormone calcitonin or CT. So, all right, so we have thyroxine and triiodothyronine, but T4 and T3. So these stimulate energy metabolism of all cells. And then calcitonin is going to tone your calcium down. It's going to inhibit a breakdown of bone, causing a decrease of blood calcium concentration. All right, so we're going to go over to, that was thyroid gland, which is the thyroid hormones. All right, so T4 and T3 are small non-steroidal hormones that are able to enter their target cell to find their receptors. And this is an exception to the general model of non-steroid action requiring an internal second messenger. So T4 and T3 influence every one of the trillions of cells in our body. They make them speed up the release of energy from nutrients. In other words, these thyroid hormones stimulate cellular metabolism. So this is going to be how quickly we can get something done. All right, and then we talked about calcitonin. It tones calcium down. An increase in calcitonin secretion quickly follows any increase in blood calcium concentration, even if it's a slight one. So it helps maintain the homeostasis of blood calcium and prevents harmful excess of calcium in blood, which is called hypercalcemia, which was in some of our questions at the end of the chapters, and she does like to pull some from those. So I would know that. All right, so parathyroid glands are small lumps of glandular epithelium, and there are usually 4 of them found in the posterior surfaces of the thyroid gland, and this is on page 241. There's a picture. It was also in the handout that we had, which I'm going to go ahead and open this up. Let's see. Okay. I'm going to go back to our chart. So the parathyroid secretes parathyroid hormone, so that's not going to be hard to remember, but it stimulates the breakdown of bone and causes an increase in blood calcium concentration. So this is the opposite of of calcitonin. I'm sorry. All right. I'm going to bump over to adrenal glands. So there's the location of adrenal glands is over each kidney, and this is also in the handout as well. Let's see if I can do that. All right, so there is a couple of regions on our adrenal glands. All right, so the adrenal cortex and adrenal medulla is what I'm looking at. And this is on this page if you have it out. All right, so adrenal cortex is going to secrete mineralocorticoid or aldosterone, which we hear that a lot. Glucocorticoids, which is cortisol or cortisone, and then androgens. So mineralocorticoid is going to regulate, I'm sorry, I just lost my place. Mineralocorticoids are going to regulate electrolyte and fluid homeostasis. Glucocorticoids are going to stimulate gluconeogenesis, causing increase in blood glucose concentration and have an anti-inflammatory, anti-immunity, anti-allergy effects. So a lot of the times when we have an illness or some kind of disease process going on, we usually get, they call them steroid packs, but that's what this is cortisone or cortisol. So then we have androgens, and they can stimulate sexual drive in the female, but have negligible effects in a male. Okay, so back to the adrenal gland. We have an adrenal medulla as well, and this produces epinephrine and norepinephrine, and it's to prolong and intensify the sympathetic nervous system during stress. So this is when you're amped up, norepinephrine and epinephrine. This is when your body's kind of in an overload state. This is when you're in your sympathetic mode, fight or flight. So let me go back. Okay. All right, so 3 different zones of layers make up the adrenal cortex, and that is the capsule, the medulla, and the cortex. And this is on page 242 at the very top. So the capsule is going to be the outermost layer, the cortex is going to be the middle, and then the medulla is going to be the most inner layer. All right, so zones of the adrenal cortex. Hormones are secreted by 3 layers of zones of the adrenal cortex called corticoids. And these are steroid hormones. So the outer layer is secreting mineralocorticoids, and in the middle is glucocorticoids. The innermost, deepest zone of the cortex secretes small amounts of sex hormones. Sex hormones secreted by the adrenal cortex resemble testosterone. So then we're going to go down to aldosterone. It's a mineralocorticoid that helps control the amount of certain mineral salts, mainly sodium chloride, in the blood. So aldosterone is the chief mineralocorticoid, and its main function is to increase the amount of sodium and decrease the amount of potassium in the blood because these changes lead to other profound changes. It works along with ADH to help keep up water content of the body in homeostatic balance, and it changes water balance and affect blood volume and pressure, kidney function, and many other functions needed for healthy survival. So then we're going to go over to cortisol or cortisone. Let's see. All right, it's a process in liver cells that converts amino acids to glycerol or to glucose. And glucocorticoids act in several ways to increase glucogenesis. They promote the breakdown of tissue proteins to amino acids, especially in muscle cells, and amino acids then form and move out of tissue cells into blood and circulate into the liver. So liver cells change them to glucose by a process of gluconeogenesis, and newly formed glucose leaves the liver cells and enters the body, enters the blood. Okay. All right, so glucocorticoids acts with these hormones from the adrenal medulla to produce an anti-inflammatory effect, and they bring about a normal recovery from inflammations produced by many kinds of agents. So, as I was saying before, a use of cortisone or hydrocortisone is to relieve skin rashes based on anti-inflammatory effect of glucocorticoids. So sometimes, if there's something going on with your skin, they'll prescribe these or like a Z-Pack. All right, so another effect produced by these are their anti-immunity, anti-allergy effect, and they bring about a decrease in the number of certain cells that produce antibodies, substances that make us immune to some factors and allergic to others. So when dangerous stimuli attack the body, they produce a set of internal responses, and this is known as stress. Injuries, physical or emotional threats, and even pollution or high external temperatures can produce stress responses. The normal adrenal cortex responds during stress by increasing the secretion of glucocorticoids. All right, so we're going to take a break right here. We have about 2 or 3 more pages left. I'm going to go get some more of my notes, and I'll be right back. I need to go get another cookie. Y'all take a break for a minute, get you a snack, get you something, and we'll be back. Let's go for like 8:10. All right, we got literally like 4 more pages, so let's bust through this. Everybody back? Everybody ready to go? Y'all have been on here for almost 5.5 hours. That's okay because we can say that we know everything because we studied. I think we're gonna do fine for real. I don't think it's gonna be as bad as we're thinking it is. I think we're overthinking a lot, and as long as we know these bolded terms and the stuff that she specifically said we know, I think we're fine. But I think a lot of us are liking the way that we want to understand it deeper. We can't just look at something and call it a day. That's not necessarily a bad thing. All right, let's roll. All right, so we stopped in the middle of cortisol. Let's see. All right, so when resisting or avoiding a threat, the increase in blood glucose can help prevent our help improve skeletal muscle function. So reduced inflammation may also help keep our muscles less swollen, so more mobile, so we can deal with a threat. All right, we're going to bump over to adrenal sex hormones. Let's see. Okay. That's androgen. So this could stimulate sexual drive in female, but have negligible effects in males. So these are secreted in the inner zone of the adrenal gland, and our male hormones, androgens, are similar to testosterone. So these hormones are secreted in small amounts in both adult male and females, and they play an early role in the development of reproductive organs. So in women, these androgens may stimulate the female sex drive, and in men, it's going to be secreted into the testes. That adrenal androgens are physiologically insignificant. All right, so the adrenal medulla is the inner portion, and it secretes the hormones epinephrine and norepinephrine, and epinephrine is also known as adrenaline. So when you think of a shot of adrenaline, that's what this is. All right, so our bodies may have ways to defend themselves against enemies that threaten their well-being. And psychologists might say that we resist stress by making many stress responses. So the adrenal medulla responds very rapidly to stress because nerve impulses conducted by sympathetic nerve fibers stimulate the adrenal medulla. When stimulated, it literally squirts epinephrine and norepinephrine into the blood. So that's pretty cool. So with glucocorticoids, these hormones may help the body to resist or avoid stress. In other words, this helps with the fight or flight response. So this is what we talked about before with the sympathetic nervous system. So it says, let's see, epinephrine prolongs and intensifies changes in body functions brought on by stimulation of sympathetic subdivision of an autonomic nervous system. So that was in chapter 9. There's a close functional relationship between the nervous and endocrine system, and perhaps it's most noticeable in the body's response to stress. So in stress conditions, the hypothalamus acts on the pituitary gland or anterior pituitary gland, to cause a release of ACTH, and that stimulates the adrenal cortex to secrete glucocorticoids. So at the same time, sympathetic subdivisions of the autonomic nervous system is stimulated along with the adrenal medulla. So that release of epinephrine and norepinephrine occurs to assist the body in responding to the stressful stimulus. So now we're going to go down to pancreatic islets, inlets, however you want to say them. And these are glucagon and insulin. So glucagon stimulates liver glycogenolysis, causing an increase in blood glucose concentration, and insulin promotes glucose entry into cells, causing a decrease in blood glucose concentration. So glucose is going to be high, insulin is going to lower it. All right, so we have pancreatic islets or islets of Langerhans, and these are merely little clumps of cells scattered like islands in a sea among the exocrine glands of the pancreas, which secrete the pancreatic digestive juice. So you have alpha cells or A cells, beta cells, and alpha cells secrete a hormone called glucagon, and beta cells secrete insulin. So glucagon accelerates a process by glycogenolysis in the liver. Glycogenolysis is a chemical process by which the glucose is stored in the liver cells and in the form of glycogen, and then it's converted to glucose. Insulin and glucagon are antagonists. So in other words, insulin decreases blood glucose concentration and glucagon increases it. Insulin is the only hormone that can decrease the blood glucose concentration. Several hormones, however, increase the glucose concentration, including glucocorticoids, growth hormone, and glucagon. So insulin decreases blood glucose by accelerating its movement out of the blood through the cell membranes and into cells. So then it's going to tell us about diabetes. So the way Ms. Putt was explaining it to us earlier, I'm trying to remember. I'm sure, Valerie, if you know an easier way to explain that, please feel free to send that in the chat, because I know she was talking to me and you when she was telling us about that. But so if your pancreas secretes too little insulin, this is going to be type 1 diabetes mellitus. Less glucose leaves the body to enter the cells, so the blood glucose increases sometimes even 2 or 3 more times the normal amount. Most cases, type 2 diabetes mellitus results from some decrease of insulin and abnormality of the insulin receptors, preventing the normal effects of insulin on its target cells and raising blood glucose levels. So screening tests for all types of diabetes mellitus rely on the fact that blood glucose levels is elevated in its condition. So, all right, so diabetes type 1 is characterized by high blood glucose concentrations and a lack of sufficient insulin. So one of these is present at birth, and then the other is going to be, as Ms. Putt said it, like a negligence in the body. So diabetes 1 is going to be little to no insulin, and a lot of people are just born that way. And then diabetes 2 is going to be like an abnormality in it, preventing it. So these people would have to have insulin pumps, and that's how they would be able to lower their blood glucose levels. All right, so type 1 diabetes is normally found in children, and the pancreas does not produce insulin. All right, so thank you, Valerie. So diabetes 1, we can think of, it could probably happen when you're 1 year old, younger. And then type 2 would be later on in your life, possibly. All right, so we're going to go over to sex glands. You have female sex glands and we have male sex glands. So the females are ovarian follicles are little pockets in which egg cells or ova develop, and ovarian follicles also secrete estrogen or the feminizing hormone. All right, give me just 1 second. All right, I'm going to bump back over to the chart, which I feel like I need to get tattooed on me before tomorrow. All right, so your ovaries are going to produce estrogens and progesterone. So estrogens promote development and maintenance of female sexual characteristics, and progesterone promotes conditions required for pregnancy. So this also speaks about corpus luteum, which is in a different chapter. And it's the process of how your body would get ready to be to carry a child. these steps are not important right now. You're not going to need to know them, but if you wanted to look at it, you could go over to the reproductive chapter towards the end of it, and it would give you more information on corpus luteum. So, all right, so your ovarian follicles secrete estrogen. Estrogen is involved in the development and maturation of the breast and external genitalia. So this is going to be involved in your puberty. It's going to give you the physical characteristics of it. The hormone is also responsible for the development of adult female body contours and initiation of menstrual cycle. All right, so corpus luteum chiefly secretes progesterone and also some estrogen, and that's like, oh, okay, it says chapter 21. All right, so male sex glands. Some of the cells of the testes produce the male sex glands called sperm. Other produce the liquid portion of the male reproductive fluid called semen. So in semen there is sperm. It's not all one thing. That gets confused a lot, but it's a fluid that helps to carry the sperm and protect it. So interstitial cells in the testes secrete the male sex hormone called testosterone directly into the blood. These cells of testes are therefore the male endocrine glands. So this is the masculinizing hormone. We'll go ahead and let you know a secret. I have an overproduction of testosterone that comes with PCOS. So that's why I have a lot of hair on my upper lip, and it's not exciting. And I have more hair on my arms than other women do, and that kind of sucks. But that's just because I have a little bit more testosterone than other women do. But it's cool. All right, so testosterone is also responsible for the maturation of external genitalia, beard growth, changes in voice at puberty, and for muscular development and body contours typical of a male. So anybody who is going through a transition, people that are transgender, if somebody is trying to transition from a female to male, they would take testosterone, and this would give them the male characteristics that they're looking for. All right, so the thymus produces the hormone thymosin. Keep going back. I just need to bookmark this really. All right. So this promotes the development of the immune system cells. So T cells, yes, we like those. All right, so thymosin is actually a group of several hormones that together play an important role in regulating the development and function of T cells, an important category of immunity agents in the body, and that's discussed in chapter 14. All right, so this small structure weighing about 20 grams plays a critical role in the body's defenses against infections and cancer. Let me let you in on a secret. I'm pretty sure they already have the cure to cancer. They're just not giving it out. But if we could make more T cells, we could cure cancer, because T cells are the key function in getting rid of cancer naturally before it starts to take over. Once it's gotten past a certain point, that's when you would get to chemo and everything. But if we could find a way to duplicate T cells, then we would have a cure for cancer. All right, so we're going to go over to placenta. This is a chorionic gonadotropin, and this produces estrogens and progesterone. So this promotes conditions required during early pregnancy. So this is getting ready. All right, so during pregnancy, it produces chorionic gonadotropins, and they're called this because they're tropic hormones secreted by cells of chorion. So during the earliest weeks of pregnancy, the kidneys secrete or excrete large amounts of chronic gonadotropins to the urine. This is discovered more than half a century ago, and it led to the development of early pregnancy tests that are still commonly used today. So this is what we're going to look for when we have a pregnancy test. It's going to pick up on those hormones and tell you yay or nay. So the outermost membrane that surrounds the embryo and fetus during development in the uterus, this is the placenta. So it produces chorionic gonadotropins, and the placenta also produces estrogen and progesterone. So this is what we use to take care of our baby before they're even born. All right, so now we're going to pineal gland. This is my favorite because I like sleep. This is considered the timekeeper, and it's like a pea size in your brain. It's really small. So this produces melatonin, which is natural in your body, and we talked about this in the nervous system chapter as well. So it's naturally produced in your body, and so it's not habit forming. So a lot of people prefer to go to the store and get this rather than a sleep aid because it's already produced in your body and it's natural. But this helps you to keep up with basically with what day it is, how the world's turning around you, because it's easy to get lost in that. So the pineal gland is easily located in a child that becomes fibrous and encrusted with calcium deposits as a person ages. The pineal gland produces a number of hormones in very small quantities, and melatonin is being the most significant. It inhibits the tropic hormones that affect the ovaries and is thought to be involved in regulating the onset of puberty and menstrual cycle in women. It's basically can be called the third eye because it's a light-sensitive ganglion cell. The pineal gland uses information regarding changing light levels to adjust its output of melatonin. So melatonin levels increase during the night and decrease during the day. And some people, I feel like mine's always on. The cyclic variation is an important timekeeping mechanism for the body's internal clock and sleep cycle. So melatonin supplements are now widely used as an aid to sleep or reprogram the sleep cycle as a treatment for jet lag. All right, so now we're going to go over to endocrine functions throughout the body. So other endocrine tissues, we're going to go back to our handy box. I'm going to go ahead and bookmark that because I keep losing it. All right, so the last ones we have is atrial natriuretic hormone, or ANH, and this regulates fluid and electrolyte homeostasis, and this is secreted by the cells in the walls of the heart. Valor has a good thing. All right, pineal, pine nut, and it's tiny. Good deal. She also added to the diabetes. So diabetes type 1 is normally found in children, and the pancreas doesn't produce insulin. Type 2, Ms. Putt said basically it's abusing the pancreas. That's the word I couldn't think of, abusing. Okay, pancreas doesn't produce enough insulin. So there's people that have this when they're born, and then there's people that end up having it later. And then they also, I don't think they mentioned it here, but there's gestational diabetes as well, and this is diabetes only during pregnancy, which is crazy. Some people end up keeping it after they have a baby. All right, so back to 247. ANH is an important regulator of fluid and electrolyte homeostasis, and it's an antagonist to aldosterone. So aldosterone stimulates the kidney to retain sodium ions and water, and ANH stimulates the loss of sodium ions and water. So they kind of balance each other out. We also have ghrelin, and it's secreted by epithelial cells lining the stomach and boosts appetite, slowing metabolism, and reduces fat burning. So it's involved in the development of obesity. And so I'm going to go ahead and say that the opposite of that will be leptin, which is secreted by fat storing cells throughout the body, and it seems to regulate how hungry or full we feel and how fat is metabolized in the body. So it says researchers are looking for how leptin works with other hormones in hopes of finding ways to treat patients with obesity, diabetes mellitus, and other disorders involving fat storage. So a fun fact about leptin is it has been researched for a long time because this will be pretty much a cure-all for obesity. It was tested in rats, and it worked in rats, but it doesn't work in people. So rats can lose all the weight they want to, and we won't be able to. But anytime you go to the store and you're trying to buy something for weight loss, I'm going to go ahead and say you're wasting your time, because unless it's got leptin in it, it ain't happening. But basically, leptin is going to tell you how full you are. So if we could find a way to trick our body with leptin, then we would be able to say that we're full and we're not. But I imagine that it's probably not possible. I just imagine that God intended it to be that way, that we didn't have it, because we'd probably be starving ourselves to death trying to look a certain way. All right, so the very bottom of this is the hormone actions in everyday organ. I'm not going to read that. That doesn't have anything bolded in it. It's not important. So definitely go back and look. I want to go back to Cushing syndrome. So tumors located in the middle zone of the cortex often result in production of abnormally large amounts of glucocorticoids. And this is Cushing syndrome, and you can see that in Figure A on page 244. And then a deficiency or hyposecretion of the adrenal cortex hormones results in a condition called Addison disease. So John F. Kennedy suffered from this disease, and it reduces cortical hormone levels due to this disease, and it causes muscle weakness, reduced blood glucose, nausea, loss of appetite, weight loss, and possibly increased skin pigmentation. All right, so that's the end of that. We made it all the way through. Thank you guys for hanging out with me. I hope this helps. I'm going to go ahead and post this, and then I'm going to go back and post the times that each chapter started. That way we know you can kind of fast forward to one if you want to go back and check it out. All right, thank you all for hanging out. I'm going to go ahead and end the stream. Does anybody have any questions before we log off? Anything that they want me to go over before? Everybody good? You're welcome. All right, y'all. I'm gonna go get me some food. If you have any questions, holler at me. I'm gonna be up super late, so. All right, y'all have a good night.